Please start the session 4, single cell phenotypic multivariate analysis, open challenges. And the first speaker comes from Institute of Cellular and of Cell Biology and Pathology, Nicolae Simionescu from Bucharest, Dr. Elena Butori, and academician Maya Simionescu, neutrophil phenotype orchestrate, healing for failure post myocardial infarction. Please, Elena.
Thank you very much. Good morning to everybody. It's a pleasure to be here today. Today I will talk about neutrophils and their changing world. This immune cells was regarded in the past as cells involved in the last phase of the immune response, with one single task engulfing and killing of microorganisms. But a lot of studies in the last years found that actually neutrophils present a large phenotypic heterogeneity and functional versatility. Therefore, it was identified different neutrophil subtypes with different roles in both health and disease associated with tissue injury or repair. And the question is, the neutrophils are cells with many roles in inflammation or several cell types. Even in circulating, it was identified different subsets of neutrophils driven by their functional heterogeneity, by maturation and aging, as well as specific factors from tissue microenvironment. Therefore, immature, mature, aged neutrophils and neutrophils with interferon-stimulated gene signature were identified in human and mouse, and each subset of neutrophils exhibits specific biomarkers. The studies that started to identify different kind of neutrophil subsets started to discover in different pathologies such as cancer in myocardial infarction. After myocardial infarction, neutrophils rapidly infiltrate the heart, where they can promote tissue healing or damages. The neutrophils can promote myocardial injury, releasing huge levels of reactive oxygen species by degranulations when a lot of proteolytic enzymes are released, such as myeloperoxidase and metalloproteases, and sure, a lot of inflammatory cytokines, chemokines, but also other means. Recently, it was found that in addition, neutrophils can form extracellular traps and extracellular vesicles and produce an increase of inflammation in cardiac injury. In the contrary, emerging evidences revealed that neutrophils are indispensable for appropriate wound healing post-MI. They can promote inflammation resolution and angiogenesis and scar formation, releasing different specialized pro-resolving mediators such as THF-beta, VEGF-A, beta-1, VEGF-A, and NXT1. Therefore, the question was which specific subset are responsible for healing or damage. And studies started to identify in specific neutrophil subsets. And in myocardial infarction in 2016, Ma and its collaborators identified two important neutrophil subsets: pro-inflammatory neutrophils and anti-inflammatory neutrophils. Pro-inflammatory neutrophils were identified in the ischemic heart of mouse model of MI. They produce a lot of pro-inflammatory cytokines and enzymatic and proteolytic enzymes, and were significant contributors to damage of left ventricle wall. Interestingly, in the day 7 post-myocardial infarction, the authors found another different subset of neutrophils, anti-inflammatory neutrophils. They express anti-inflammatory molecules and help for inflammation resolution. Following a strategy similar to that for obtaining inflammatory and anti-inflammatory macrophages, the authors obtained in vitro these two neutrophil subsets, inflammatory and anti-inflammatory subsets. 4 years later, combining single-cell analysis and surface epitope technology, other group identified at day 1, 3, and 5 post-MI, and also in the ischemic tissue from MI mice, 6 different clusters of neutrophils with specific and time-dependent patterning and proportions. Importantly, at day 4, a major subset of Siglec F high neutrophils were with a unique signature was found within the ischemic cardiac tissues. These neutrophils that are expressed high level with Siglec F high are specifically for this tissue and are enriched for different inflammatory molecules such as cell adhesion molecules, ICAM1 or cytokine TNF-alpha, and functionally are characterized by increased ROS production. 1 year later, or maybe in the same time, who knows, another group using single-cell transcriptomic landscape of the neutrophils, also in a mice model of MI, identified five different major subsets of neutrophils, and importantly, a dynamic pro-inflammatory cardiac neutrophil, a Siglec F high positive, that were represented 25% of neutrophils, of all neutrophils in the first day, and the number increased in the day 4 at 50%. This subset neutrophils were also enriched for gene associated with oxidative phosphorylation, such the previous group found. Deeper characterization of this subset of neutrophils found that they express a lot of inflammatory molecules, including the TNF-alpha and ICAM1 that was found previously. And regarding the signaling mechanism, they were found associated with MIC and NFKB signaling pathways. All this data suggests that Siglec F high neutrophil subset found in the ischemic heart has an intense pro-inflammatory status. In this context, our hypothesis was that different population of neutrophils may have specific function that can predict the disease evolution post-MI. And our aim was to perform a complex analysis between inflammatory and anti-inflammatory neutrophils population to find their functionality in order to identify molecules or population of neutrophils as markers for disease evolution post-MI. As experimental design, we isolated mouse neutrophils from tibia and femur. We also have had human neutrophils isolated from circulating blood from patients with acute myocardial infarctions infarction. The blood was recorded in the first 24 hours post-MI. The mouse neutrophils were polarized toward the pro-inflammatory N1 neutrophils or anti-inflammatory N2 neutrophils using LPS and interferon gamma or interleukin 4 for 2 or 18 hours. Our control neutrophils, inflammatory neutrophils, or anti-inflammatory neutrophils were subjected to RNA sequencing, and our data revealed distinct transcriptomic profile of these three populations, with N1 neutrophils upregulating among 800 genes and downregulating 195, and two neutrophils upregulating around 500 genes and downregulating 130. Between the inflammatory molecules upregulated in the N1 neutrophils, there was a lot of cytokine and the chemokine and the selectin is here. Our validation studies found that the gene expression of different inflammatory molecules, cytokines, TNF-alpha, interleukin 12, interleukin 1 beta, interleukin 6, but also chemokines CCL3, CCL5, and CCL2 were significantly increased in inflammatory neutrophils at both times, 2 hours or 18 hours, not in anti-inflammatory neutrophils. Between these molecules, the previously markers associated with Siglec-F high neutrophils were also increased. Enrichment analysis of the gene cluster of highly upregulated genes in inflammatory versus control neutrophils identified as most upregulated signaling pathways in N1 neutrophils, TNF signaling pathway and NF-KB signaling pathway, similar with those found in enriched Siglec-Fi subset. Moreover, the inflammatory response was the most. Here are the signaling pathways, and here is the inflammatory response. Was the most upregulated biological process in inflammatory neutrophils. Our data suggests that there is a higher similarity between our inflammatory neutrophils or polarized in vitro with the Siglec-F high neutrophil subset found in the ischemic heart post-MI. Next we question if this inflammatory phenotype of neutrophils there are also in human patients post-MI. And we started a comparative study between mouse neutrophils and human neutrophils isolated in the first 24 hours post-MI. And our data show that the inflammatory molecules, interleukin 1 beta, CCL3, ECAM1, and S100A9 was also significantly increased in human neutrophil post-MI, as in N1 neutrophils polarized in vitro. S100A9 is important alarmin that it's found as a dimer with S100A8, and this dimer is a promising biomarker and therapeutic target in cardiovascular pathologies. Therefore, plasma levels of this dimer was found highly increased during the ischemic event in acute coronary syndrome or in other cardiovascular pathologies. The dimer binds to TLR4 receptor and prime the activation of inflammasome that leads to secretion of interleukin-1 beta, which further interact with specific receptor on hematopoietic stem and progenitor cells from bone marrow to stimulate myelopoiesis in circulation. Recently, the Professor Stoffel group, our collaborator, found that short-term blockade of S189 using a specific blocker, ABR238901, decrease mobilization of monocytes and neutrophils in the infarcted area, leading to inflammation resolution and therefore to improve function of improve cardiac function post-MI. To further investigate the beneficial effects of this inhibitor, we look at effects of ABR on the inflammatory and anti-inflammatory neutrophils, and we found that polarization of neutrophils in the presence of the S189 inhibitor do not affect the cytokine levels, but significantly reduce the chemokines levels, the chemokines CCR3, MIP1 alpha, arantes, and MCP1 at both investigated times. Therefore, in addition to reducing the myelopoiesis in the myocardium, this inhibitor reduces the expression of chemokines produced by inflammatory neutrophils. The gene expression data was confirmed at protein level. This inhibitor significantly decreased the secretion of these chemokines as soluble form of these chemokines as identified using cytokine profiler array or ELISA array. As a result of this decreased expression of chemokines, the neutrophil transmigration and important function of neutrophils was significantly reduced. Other important neutrophil function, reactive oxygen species, that is essential for efficient microbe killing and degradation, but excessive reactive oxygen production induce dysfunctional signaling and mediate oxidative damage to proteins, amino acids, and lipids. In myocardial infarction, reactive oxygen species stimulate the release of pro-inflammatory factors in the ischemic myocardium. In our experiments, using seahorse technology, we found that oxidative bursts of inflammatory neutrophils and of human neutrophils post-MI was significantly increased as compared with control or anti-inflammatory neutrophils. A feature similar with ischemic cyclic FI neutrophils. As a consequence, the reactive oxygen species was significantly increased in both inflammatory neutrophils or MI neutrophils. Neutrophil degranulation is an important step for regulative function of neutrophils. Myeloperoxidase is released by primary granules of neutrophils. It's an important inflammatory marker elevated in acute myocardial infarction, and the plasma concentration of this enzyme predict mortality post-MI. Our data show that inflammatory mouse neutrophils exhibit increased activity of increased levels of myeloperoxidase. Also, previously it was found that myeloperoxidase is increased in patients with acute myocardial infarction compared with patients with stable angina or controls. Specific inhibitor of S189 significantly reduced the myeloperoxidase. Metalloproteases are important proteolytic enzyme, and post-MI increased levels of MMP9, degrade extracellular matrix and promote leukocyte infiltration. We found that metalloprotease 9 activity was significantly increased in inflammatory neutrophils, but not in anti-inflammatory neutrophils, and that the inhibitor reduced significantly this activity. In human neutrophils, we found that gene expression of MMP9 and also plasma levels were significantly increased at neutrophils from MA patients. Elastase, it's produced also from primary granules. It's an enzyme that degrades elastin, collagens, and fibrinogens. It is released in early stages of ischemia, and its inhibition has been shown to reduce infarct size. We found also increased activity of elastase in inflammatory neutrophils, but not in anti-inflammatory neutrophils, and also in the myocardial neutrophils. Regarding the signaling mechanism responsible for activation of inflammatory neutrophils, our NANSIC data found over 20 different signaling pathways that were activated in inflammatory neutrophils versus control, and between them the most signaling pathways with all around 40 differentially expressed molecules were NFKB and TNF signaling pathway, as already mentioned. Between these modified molecules, we choose to investigate three of them, three most important. P65 subunit of NFKB signaling pathways, the ERK1/2 MAP kinase, and protein kinase C. Our Western blot data show that the phosphorylated form of P65 was significantly increased in inflammatory neutrophils and not in anti-inflammatory neutrophils, and S189 blocker significantly reduced. Moreover, ERK1/2 was also increased in N1. No significant differences were obtained for protein kinase C. In conclusion, our data show that neutrophils are transcriptionally dynamic to response to injury. Circulating human neutrophils in the first 24 hours exhibits the phenotypic and functional similarity with inflammatory neutrophils subtype and with Siglec-FI subset found post-MI in a mouse model of myocardial infarction. The function of these inflammatory neutrophils are detrimental for myocardial repair for post-MI, and the pharmacological blockade of S189 dampens the function of the pro-inflammatory neutrophils, promoting the albumin as a target for therapeutic intervention in inflammatory disease. Some of these data were published recently in a paper, and I want to thank to my colleagues from laboratory, thanks to Maya, because they allow me to work in this very interesting project. To my colleagues from the lab, Ella, Anka, Andrea, Rezvan, Ilana, and Letizia. Thanks our collaborators that provided us the blood for patient with MI, Dr. Elena Barber and Professor Fredan Belenescu, and our collaborators from Lund University, Sweden, Alexander Shkyopo and Gabriel Jacobson, and also thanks this data were possible due to great project, PCCF project that will finish this year. And thank you for your attention. Thank you.
Thank you very much for your very nice presentation. Open for questions or comments. Please, Greg.
I have one question concerning pro-inflammatory cells. How they actually get signal to go to the infection place in the heart? I mean, in principle, as I understand, because I'm not in that field, so somehow they have to get signal and accumulate there, and then they do the function of clear air up the tissue. Is that correct statement?
Yes.
So what are the signals that would get them there and then activate what you very nicely described, you know, the degradation function and then increases, I suppose, inflammation?
Yes. Immediately after infarct, actually there is the oxygen supply, the blood is not coming in the yes, in the heart, in the left ventricles, and there is a massive damage of the cardiomyocyte cells. They that and produce a lot of alarming damps and alarmins that are released and signal in circulation for myelopoiesis. I showed with yes. And there is a huge myelopoiesis and increased levels of neutrophils. They are forced that come in the tissue, in the injured tissue post-MI. Actually, the number of neutrophils for infarct predict the worse or better evolution of the patients, with a huge number worse evolution of the patient post-infarct.
So somehow if it overdoes this, it will be not for the benefit of the patient. It has to find some kind of balance.
Yes.
Is that an understanding? Because
yes.
it's like overshooting the reaction, and then that it is then damaging even more the tissue, then it should be sort of clearing it up. I mean, that's the whole, I take that as
the inflammation.
Story.
The inflammation is very benefit because it start the resolution inflammation, but only to be limited to the first 3-4 days, and to allow the anti-inflammatory factors starting to 5 days to become and to promote scar formation and inflammation resolution for a better result.
So this is in therapies actually one should consider these two things, not to too much suppress or not to, so to find this fine kind of physiological tuning balance. I think that's the key
thing. Actually.
In every therapy, by the way, this is the case, I suppose.
Actually, the inhibitor I talk about had a very nice effect, but only if it was used in the first days.
Yes.
But when our collaborators give these inhibitors to mice for a longer time, it works the improvement.
Then the tissue damage is even then or no clearing up.
Yes.
I enjoy also your this single cell analysis with the RNA. I thought we will continue a little bit, but in other direction.
Yeah.
Thank you.
Other question? I would like to ask you something. What happens with metalloproteases from extracellular matrix after MI?
What?
Because you mentioned metalloproteases after the granulation of neutrophils.
They are released. Yes, they are released in the tissue, and depending of their levels, sure they can help in a first step. But if they also, as with the inflammation, if there are a huge expression of this metalloprotease, this scar formation can not start to form, and this will worsen.
Yeah, it's a cascade.
Yes, it's cascade.
Yes. Yeah. Thank you once again. And now I would like to invite Professor Vladimir Mislevich from Basel, Switzerland and Nanjing, China. Please. 30 minutes, please.
Yeah. So we will continue now in the single cell analysis. This is the session, right? And what I would like to talk a little bit more general and about our voyage towards actually single cell analysis with a single molecular detection. We are far from that, just to give you the warning before that. Reaching this goal, and it will probably take several years, I hope several years, probably more, due to technologies, development actually machines also, and our ingenuity to be more creative than until now. And I would like to express actually also my happiness that I'm here in this multidisciplinary kind of audience. I learned a lot of things and what really inspired, and I thank to the Michaela and Eugen for inviting me here and to really enjoy this nice atmosphere. So I'm actually associated with my own company, which is a single person myself, and we are actually working in many different topics, and one of these is that. And as well, I have really a pleasure to, together with Tim Hunt, to create the research institute with a legal company in China, and also I'm associated and affiliated with Nanjing Medical University, where I teach immunology. So, okay, so this is the first question, to what I would like to raise, which is kind of introductory, and this is how many cells are actually in a human body, because we want to analyze the single cells. And according to the estimates based on the many different analyses, and there are several papers published, actually few papers published, there are about 30 to 40 trillion cells in a human body. These are enormous number, right? So we want to do the single cell analysis. First of all, it would be destructive, right? You would like to analyze everything in order to make conclusions. So it's practically, as I say, impossible in that sense. You can do post-mortem something, but this is an enormous thing. This is one of the somehow obstacles which will be difficult ever to solve. And the second part is how many different cell types there are, and how do we define the cell types in the human body or any kind of the living organism, multicellular organisms. So in humans, it is again estimated that there are about 400 different cell types, and they are here presented in a different type of the graphs by the number of cell types in the human body and the mass that they occupy. These are very different. As you see that in this graph, the majority, 80%, are actually erythrocytes. But if you look at the mass, because they are small cells, then you see that they are actually kind of the minor cell type. And this raises also the second question of how do we define the cell types. And this is the reason why one should do the single cell analysis with single molecular detection in order to be able to really then truly say what is a different cell type. And in addition, you have to do that in a time scale, because the cells change, as we see there. According to the external stimuli in any organism, the cell change metabolically. And where is the limit where you say this is one cell type because it expresses different genes and when is another type? The classical way was morphological, which is an excellent one, because you simply see the cell and this is the result of all gene expression, which are there, and that means expression of the proteins, glycans, RNA obviously, and lipids and glycolipids. So this is really kind of fundamental question which one has to bear in mind when he's asking the question of the single cell analysis, the single molecular detection. So then the second thing is how many molecules are in a cell, and this will differ depending on the size of the cell, that means on their mass, what I showed you before, and it will depend naturally on the technique that we are using to detect, and not only detect, but also to quantify each molecule in each cell. So this is just a graph which is showing you the major biopolymers, as well as the metabolites like small organic molecules, as well as inorganic molecules. You should analyze everything in that sense. And it shows you two different things: how many molecules are present per cell, and obviously, as I say, this is a schematics, very much generalized, because it depends on the cell type, what we so far know. And it shows also how much micrograms, or in this say actually picograms, is present per each cell. And this is showing the detection limit, and this is the detection limit of the mass spectrometers, which is so far the most sensitive way of analyzing biopolymers or organic molecules or even ions. So you see that this kind of shadowed or glassy shadowed line is showing that anything below that would not be able to detect. So luckily for the nucleic acids, so DNA, mRNA, it was in the last 20 years developed the amplification method. So even the RNA here is, if you see in amount, in the picograms, is the smallest one. You can amplify it so much that this is so far the best technology developed so far for analyzing single cells, what you have just presented. And this is really the marker now which people can define which cell type and what is physiological state of them. It's incredible how many publications have come out in that field just in a couple of the years. And technology is just superb. But there are many problems associated. Whenever you amplify anything, you really, and you go from the single cell, you have to know for each molecule, is the amplification rate same or not? And it's very difficult to do the controls for that. So this is one of the challenges for any kind of the amplification. Do you amplify each type of the molecule exactly the same according to their concentration or not, or according even to their structure? Because these are really, you cannot check or do the controls very easily with that. Okay, so let us just move then to see that if you have another kind of imaginatory graphical presentation. So any molecule, it doesn't matter now which one you are analyzing, with or without amplification, because you cannot amplify really the number of the molecule of protein or glycans or lipids. Then this line, which is the line of detection of the mass spectrometer, which is improving always more and more, is showing that actually you will not see the molecules are below detection. So that means if you analyze now this population of the cells, you will be missing a lot of information. That's why single cell analysis is very important in many different areas of the biology and medicine. Okay, so this is bringing us to the question why we want single cell omic analysis with single molecular detection. So, as it's sort of summarized here, the nature and the extent of cell diversity are its own problem in the biology. We really don't know, even in the timeline of the normal adult, it means without differentiation, without anything, how the environment changes actually the cell metabolism, to what degree it changes, and this is then how the pathology will develop according to the external stimuli. So, as you see here, we have this in cell biology, microbiology. One of the key questions is stem cell differentiation, de-differentiation, differentiation, regeneration connected to it, then stochastic epigenetic variation. In immunology, this is very important. We are surviving because apparently, what we believe, there are stochastic or random changes in the genome with a combination which allow production of the variety of T cell receptors or B cell receptors, which are then afterwards the antibodies. And then this is again connected more in biomedicine, very important parts, transfusion and transplantation, then analysis of microbiopsy, which also leads to early detection of the cancer. This is especially very important field. analyzing the single cells. Microbiology, another which appeared here, and parasitology, then as I said, immunology connected with the diseases, and as well as the drug resistance, which we can discuss as well in the yesterday's sessions. So how do we do or how should we do, or how could we do in the future the single cell and single molecular omics? That means detecting every single molecule in every single individual cells. And this obviously is giving us the answer we have to develop a nanobiosensor. That would involve developing the type of the chip that you can put many different cell types, and then using this chip to put into some kind of detector. This should be the best mass spectrometer, which could then detect every single molecule. I mean, and as you see, the mass spectrometer can actually not so far detect a single molecule. There are different techniques. You can detect individual single molecules, but can you detect all of them within one cell with the diversity of biopolymers and diversity of organic or inorganic substances? And then now, as you see, this is our travel towards that, which is, we don't see actually the way still how it could be done. But I will show you what we have sort of tried to do, some multidisciplinary group and several of my colleagues and friends, at least the ideas how we could go towards such objectives. So, so what are the necessary procedure steps? The number one, if we have a single cells, we have to separate single cells. It's the best thing is the blood, right? You have a blood and then the cells are not adhered. If you have cells adhered in the tissue, you have to de-adhere them. As the moment you de-adhere, you change the environment, the molecules change and so on. Even if you don't use the enzymes, you cannot control it. So this is the first very difficult problem. So then on the molecular level, again, you have to separate each of these molecules that you can make some kind of the quantitative number, how many different type of molecules. And in order to do that, by detecting, you have to use a mass spectrometer, and then it will allow you to quantify and as well to identify which type of the molecule. By separating, you can actually also go to the identification. Ideally, you would have to sequence every molecule, but you can use also indirect method or using the different standards, separating and then identification would be held in that, to help the identification. So, and if you think about just the polymer, the major things are protein, glycans and metabolites from a single cell. We know that RNA and DNA we can amplify. So this remains the major thing in a single cell analysis, has not yet reached the level of the DNA and RNA, nucleic acids. So, so there is the direct method, that means would separate, and you have to use the array where you put the single cells and then separate from each single cell all of these biopolymers. And what we have thought probably the best way would be to use kind of nanoelectrophoresis, which I will show you the chips that we designed, and that would be then the indirect method, which could use the antibody or any kind of detector for different type of the biopolymers. That would be array device. So we call it nano in microarray. That means microarray would accommodate a single cell, and with each micro dot you would have a nano array, which would then accommodate a different type of the antibodies. Let's put or aptamers or phage display library kind of things that would be able to detect different type of the molecules. So I will show you now here how actually we constructed this. Okay, to show you the movie how we actually try to construct this. And this is just an indication about the method which is used. This is a focus ion beam, which is now engraving in a nanometer scale the letters. Oops, I pressed it the wrong button, so we go backwards. We go backwards. Okay. So what we are doing now, this is how to construct with the focus ion beam, which was developed by the colleagues and friends in Orsay Physics in France, with whom I collaborated for many years. Gerard Ben Assayag, with whom I worked for many years, who developed some of these actually instruments. What you see there, that in a lifetime you can engrave the nanoscale any object that you want, and at the same time with scanning electron microscope you can actually follow what you are doing. So making the prototypes, this is the method that you can use, or probably the only one. If you produce mass production, you can use e-beam lithography or actually other type of lithography techniques to construct practically anything. But this is for prototypes, and what you can see here, you can do many different things. So you can engrave, and you can also monitor that. You can engrave different shapes. These are kind of electrophoretic path where the molecules will be put on the beginning and actually can have a long path in order to separate. And in a moment you will understand better now what we mean with that. So this is another now kind of the construction, how we do the construction of these nanoelectrophoretic devices. So first we start to engrave the chamber where we will put a single cell. This is 10 by 10 micrometers, which will accommodate one cell. Then we deposit with the same machine the electrode. This is gold or platinum, because the same instrument can deposit. Then we are again etching with the gallium ions the part where, so this will be outside. On the top you will see in a moment the schematics, and we are now further engraving and making the channel, the place where will be put cells, and then it will be the channel growing here where you will be electrophoretic path for separating different type of the proteins. So this is very nice, I mean, to see how it really works in the right. So you can see these are kind of in a nanometer scale, you can control practically everything. So this is the final product, or the prototype, where you can see here we have engraved the chamber where you can put a single cell. This is the channel now, which looks very small, and these are the electrons. So the scale is here, and then what you can see now, when you start to calculation how big should be that, this is 200 nanometers, this is probably 2 nanometers, how deep it can be. What we do is actually we engrave the part of the, this is silicon, and what we do before that, we coat this, before engraving, with the lipids, with the self-assembly layer, then we engrave, and we have exposed the hydrophilic part. Here is the hydrophobic part. When you fill this chamber, practically you will have like an open vein of the channel, which will allow you now to have very little contact on the interface to the silicon or whatever you can call that, polyethylene glycol, whatever you want to have hydrophilic part, so that you don't have in such a small things effect of the walls, which will not allow you to do any kind of separation when you have a clogging. So now what we can do is practically we can construct, these are the processes, many different type of the shapes, deeper, so which can be tested for different type of the cells, or different type of standard proteins in order to be separated. Now, unfortunately, this is a disappointment maybe for two. We could never still do this real separation. We could only on the bigger one due to many different challenges. But what we have found is that within 1 minute, you can, if you have the array, you will build up the array of this sort of nanoelectrophoretic parts. Oops, up to 1,000 of them you will have. And so if you put 1,000 of the cells within 1 minute, you will get electrophoretic separation of practically, let's say, protein of the glycans, which are easy to analyze. And now we come to the, okay, so you can do in a parallel as well as you show here. And as, I will come to this point, as for the detection of this, I will tell in a moment how we will detect this. But before that, I want to say the second part of the analysis. which is nano and micro array devices. And the same principle of the construction can be done that you have micro, 10 about micrometers wells, and in each of these wells you will then engrave then a nanoscale wells where you can put now your antibodies or aptamer or peptides from phage display library, which would be capturing then the proteins from the lyse single cells put in this array. You can accommodate about 2,500 cells on 1 centimeter microchip. So how they look like this, again, this is showing different sizes and different types of the, this is a microvel. In each microvel you can put, again, as I say, many nanovels that you can put the molecules. Obviously you can see this is a very difficult technological part which has to be further developed. How do you put each of these molecules in each of these wells? How you control hydrophobicity, hydrophilicity? But part of these are relatively easy with self-assembly layers and so, but the parts are difficult because it would take a long time to position with AFM, for example, each of these molecules in each of these probes. But there are some solutions we which tried to develop since 10 years, but we didn't advance so far much. So now we come to the point, the last part of the talk, how can you detect now these molecules, right? Because you detect a single one. So I told you mass spectrometry, it's kind of difficult. What would you do with the mass spectrometer? You have to release them from there or to take, so that's no way to do it. So there is an instrument which you heard probably, it's called the secondary ion mass spectrometry. And this is the nanosims, this is the dynamic sims, very complicated instrument, 1.5 million, has to work in a clean room. And in principle, it is actually a mass spectrometer with, which collects the secondary ions after the primary ion beam hits the sample, and like atomic bomb, practically take everything what is on the surface of our chip or our interferotic guide, and it can scan. It is a scanning instrument. So by every 30 nanometers, it would actually scan, take everything out of it, put in a mass spectrometer, and detect. So this is a nanosims, and this is a time now flight to detect, and it shows from this Professor Arlinghaus in Münster, with whom we had also collaboration, and he's one of the experts in the so-called static SIS, where you can obtain even a spectral analysis of the fragments of the molecules that are there. And the first one, you get actually atomic atom composition of the molecules. So this is the schematic part, which again shows if you have a section of the cell, let's say, like for an electron microscope, and you put it in the SIMS and then you scan, and it is shown here, so showing that this different atom, let's say of one polymer, which is for example in the membrane, so what you can detect different elements, and each of the element with this very high resolution of now to 30 nanometers will show where it is positioned. This was labeled the toxin with N, I think it was labeled with N15, is showing actually on the membrane in green here. So each of these is actually the map of a different, I think this was a phosphate, so you see it is nucleic acids and nucleolus, I think, was accumulated into the nucleus, should be in a plasma membrane, but it was fixed to extract the phospholipid, so actually you don't see it there. And so different type of atoms or elements you can see with this with a very high precision, because it has a 30 nanometer resolution. So we want to see now how much you can go down in a quantification. And I took a very simple approach, mixing up polyethylene glycol and the BSA, and you make a serial dilution of this, and you just look for the sulfur. You can look for all of the elements, and what you can see by the dilution that practically you can get to the single molecules, and these are calculations, not to go into details. You can practically visualize and detect a single molecule of the BSA just based on the sulfur. So now you can ask the question, why this is a good method compared with the classical mass spectrometry? Very simple answer. In the single molecule detection, you have a signal of one molecule hitting a detector. When you split up the molecule, which is done also in LSMS, by the way, and then increase the sensitivity, instead of one signal, comparing to the noise, you will get 100 of the molecules. So if you have more atoms, your sensitivity will be there. So if you detect now a single molecule, it's thanks to several same of atoms present in that molecule. So, and this is now showing to combination of that, of the previous experiment, to see how deep, because it's a 3D measure analysis, you can scan and scan again and scan again, and each of these is now showing on AFM, take the picture of, take the sample out of the SIMS after the few scans, and then analyze it with AFM again, and then you see that you can clean up all of the BSA, and bringing you actually practically complicated picture for each element. You can actually follow exactly in a 3D, and then you can calculate exactly in your nanoarray what and how many molecules you have. So to get to the conclusion, single cell analysis, our approach, which will give you the high sensitivity, two types, separation method, we have separation method and indirect method nano and microarray. SIMS and scanning probe microscopy are the solution for that. Thanks, and
Thank you very much. Question, comments?
This is obviously amazing, and I want to ask a simple question: why not just go directly for the tissue? So do what you just showed at the end. Why rather purify all the elements? Simply scan with the electron beam, I guess, the surface of whatever preparation you inquire, and then you get your molecules.
That is,
and I propose a tissue for that.
No, right. This is that's why I show you the cells. These are not our results. These are again results in collaboration that we were involved. This is from the literature. So with the University of Rouen and in Paris also, and then Orsay Physics. So there is a problem with that, because what you do in a dynamic synthesis, show you have actually analysis of the elements. So if you do not label the molecule with isotope, with a heavy isotope, you do not know what you are looking. If you look with a static mode, using usually they use the time of the flight, you will get fragments. Yes. We tried to do that long ago with Professor Arlinghaus, and he tried it many times. But the spectra are so complicated so far. They are much more complicated than if you do like an SMS or this. So very difficult to actually identify what are the molecules that you are looking. Okay. The possibility that you say is a combination, as is a nano in micro arrays, that if you have developed many more probes, which is again a very big problem, right? Then you could actually sort of labeling, and that will enable identification and maybe detection. What people are doing, it is not the nano SIMS, but what we have heard before is using actually just transfer of using the mass spectrometer that would scan in a micrometer range. So you have a bigger spot size, you get more atoms, and then you can sort of analyze. This is usually used for the tissue analysis. So in a way, we are in a problem with that. But that would be the ideal case.
Yeah.
What about the water handling in these very small volumes? Water handling, I mean, it's a very big problem.
Yes, that's why what I wanted to say, but shortly I mentioned this. Usually you coat the chip with lipids, then you engrave that. In lithography, you can also do this kind of things, right? And then this will be hydrophilic part. You can also modify this hydrophilic part. You add the water, you have to keep it under the pressure and the humidity, so there is no evaporation and sublimation. We made such a chambers, okay? They're difficult or not, you can do that, okay? And then after you finish the electrophoresis, actually you just dry, because you have to go into high vacuum in order to analyze the secondary ions by the sinks. So practically you dry and you separate everything, and then you can actually start to scan and then get position. Depending on the position, you will have each molecular spectrum of that. So the water is a problem, yes, but
the problem is to manipulate so small volumes in the scale of pico and femto.
that's right. These are what I say. We tried this I don't know, 12 years or so, but the problems are also obvious in the funding and the interest of the industry, because they still see it's too far. Obviously, it is too far. But, so you need the people in technology to engineers.
Eugen, please.
Vladimir, just a stupid question from my part. It's a dream in a dream.
Yes.
So what you do is what you
Trying to
what
do
you're
that.
trying to do and are going to do is fantastic. Is to get a multi-dimensional picture of a cell at one moment.
Yes.
Can we have the time lapse, the dynamics of this multi-dimensional
I always make that it's not a joke, but it is. I learned it as a student when I was in Woods Hole, Marine Biological Laboratory. They say biologists are smash and spin. Okay, so, and then I was also asked by many people, also by Emmanuel, it's like, we are really distracting, you know? I mean, we sort of and then we analyze. And the wonderful thing in the beginning was from the Professor Hell, who made the talk, when we also talk about dynamics. I mean, a live thing is the really, so we cannot, I mean, my home lesson is, and what we discussed many times also in Dr. We must combine some, you have to have a live dynamics, but knowing the kind of catalog of this, which is it important or not? Yes, it is important. It looks trivial at the end, you know, but it's important. So combining only this, that's the way how to solve. Because dynamic in a single cell analysis, what you also presented before, is the key thing, right? Because change the temperature, one of my epithelial cells which is more exposed is going to react differently than the other, right? So how can I do it? I mean, you have to stop and so. This is, and then you have to take time lapse. This is the dream, but combination, I think I see it only as a combined approach, is that you combine the knowledge and somehow maybe advance one little step. But these are really challenging and fantastic things which enrich our life, is sort of thinking and trying to move the frontier of the knowledge, right?
Yeah. Okay, because the planes. Okay.
Thanks.
Thank you. Gabi, please.
All right, good morning everybody. I'm Gabriel Professor. I'll be chairing this session on single cell dynamics, and we'll start with an exciting talk from the Polytechnic Institute by Mona Mihailescu, and it's about digital photography and application to cell dynamics and cell cycle.
Hello, I'm Mona Mihailescu from Polytechnic University of Bucharest, and first of all I want to thank the organizer to invite me here. And also I want to say that I am glad that organizers invite here fathers of digital photography microscopy. I am glad to see here Bjorn Kemper and Pedro Ferraro. I learned a lot from your articles when I started my laboratory. Now I am present our journey in the cell's life using digital holographic microscopy, and I want to convince you about their advantages, its advantages. We started our laboratory and we built our experimental setup from components. The setup is based on the Mach-Zehnder interferometer in off-axis configuration, in transmission because all biological samples are transparent. We work with different components. We have great resolution CCD or very fast MOS camera, photon, to record fast processes. This is the step when we record the holograms. Here is one example. And then this step is followed by the reconstruction step when we use commercial software from Linsetec, a company from Switzerland, to reconstruct the 3D image of the cells and to find the phase shift of the in every pixel of the cell. Here is an example of quantitative phase image. This is the result of any reconstruction process. It is quantitative because in every pixel we have information about the phase shifts introduced by the sample in the optical path. And this phase shift is proportional factor with refracting index with factor is proportional with the protein content. In last years, we collaborate with a company from Magurele, which has the commercial device for digital holographic microscopy, also from the same company, Linsetec. And the last studies were we done with this device. Okay, digital holographic microscopy are not capable to see a single molecule as in the previous presentation, but it has different advantages. It is a label-free technique, are commercial devices to see the cells inside the incubators, and it is the most fastest technique. We can monitor fast processes in real time. The speed is linked only with the CCD camera speed. And the great advantage is that we have information in the three axes, also in the axial dimension. Disadvantages is the lateral resolution only in micron scale, but temporal and axial resolution is good, very good. Based on these advantages, several applications are in literature to study dynamic processes like drugs' effect on living cells or tomographic flow in cytometry, tracking of cells and bacteria, their trajectory, or cellular activity. Our first direction to develop in our laboratory was based on studies to meet a need for Institute of Laser Plasma and Radiation Physics from Magurele. They made some substrates by laser direct writing via 2-photon polymerization. and they wanted to see these substrates and cells to adhere on these substrates. These samples were very complicated for our technique because they are too high, are not so transparent, and they are multi-layers. And for this we use different liquids to immerse these samples to made that refractive index of the liquids be very, very appropriate with the refractive index of the substrates. For this, we applied the decoupling procedure from Rafraz when we immersed the polymers, the substrates, in two liquids with very appropriate refractive index, and record two maps of phase, and to obtain separated the high and the refractive index in every pixel XY from the plane of substrate. On these substrates, we monitor the cells which grows inside them. In a few days, we have after 1 day, 3 days, and 7 days, and we can see that the cells grow near the walls. And after 6 days, their colonies is in this mode, and we monitor only simple parameters like maximum height, major axis, minor axis, or footprint area for these samples. Another complicated sample was these micro reservoirs. Because they wanted to fill these reservoirs with drugs and to monitor their control delivery. And we made some reconstructions for simple substrates, and with substrates where the cells grows in different mode. Substrates, the macro reservoirs have a different distance between them. Our recent study about substrates wanted to prove that digital photographic microscopy is capable to distinguish between regions with different materials in the same substrate. And here is a substrate like a chessboard with each square is from a polymer or a polymer with nanoparticles. But the great advantage of the digital photographic microscopy is that it delivers phase shift in every pixel from the cell, and this is a quantitative value to compute many parameters with the main scope to characterize and identify the different cells from a great population. worked with local parameters like refractive index and the height of the cell in every pixel, and global parameters like geometrical gradient, fractal descriptors, modal coefficients, autocorrelation function, optical phase shift, entropy, dry mass, and dry mass density. This is the most good results that from phase shift we can calculate the mass from phase to mass values. For automatic classification of blood cells from a population with different types, the first study was to compute gradient from the cell's centroid. But first of all, our MATLAB code wanted to identify separately each centroid of each cell. From the original reconstructed image, this is impossible because some cells are very close, and after some procedure to enhance the contrast and convolution with Gaussian filters, the cells are identified separately, and we computed the gradient from their centroid because the mature red blood cells have this minimum in center, and then the phase shift increase, and for immature red blood cells we have this maximum in center, and then the phase shift decrease. We computed also the volume, and these results were done in collaboration with Oncology Institute from Bucharest, and they made also analysis in flow cytometry to confirm our results. Another study was to monitor cells in different stages of deformability. We made some simulations where in a population with different cells, we made some discs as ellipses shapes, and analysis to classify the different deformability state was to monitor curves in curves of isoamplitude in reciprocal space here, and experimental to have cells in different deformability state. We insert them in a solution with different osmolarity, and parameters to monitor them was eccentricity, minor radius, and sphericity coefficient. Because for example, in hypotonic solution, the minimum of red blood cells goes up. We go further to find some criteria for automatic classification of cervical cells. This collaboration was with Lotus Hospital from Ploiești, and they give us some samples with normal and abnormal cervical cells. First study was to see the face profile, and for normal cells the profiles and the face profiles and the histograms associated. Here we have bimodal histograms, and for abnormal cells we have this abnormal distribution. In collaboration with computer science faculty, we developed a code to fill the volume and surface, taking the advantage that digital photographic microscopy deliver three-dimensional shapes, and we fill these shapes with balls with different radius, and compute the fractal dimension, and when we represent their evolution, we can see that have different slopes of the at a small radius and different slopes and of big radius, and different distribution of, in surface and in volume. Like this, as distance to compute the difference between them, we employ the Bhattacharya distance based on the mean and standard deviation values, and we find that distances are bigger at volume small scale and surface big scale. Going further to study the difference between cells, from now on I will present studies made in collaboration with University of Medicine from Bucharest and their collaborators from hospitals to find criteria for cell malignancy level. We employed murine melanoma cells in a perfusion chamber. We need this perfusion chamber because here we also apply the decoupling procedure to separate the information about the high and the refractive index of the cell. In bright field microscopy, two malignity levels F1, and F10, the images in bright field microscopy are similar, but in digital photography microscopy, the histograms of phase shift are different for cells with low malignity potential and for cells with high malignity potential. And also the refractive index is statistically different from these two cells category. The refractive index was computed after the decoupling procedure. Also, the results are validated by these two techniques in as biochemical analysis. Further, the colleagues from medical university wanted to differentiate the cells from their malignancy level and also from the cell cycle. From this reason, they use the protocol to fix the cell in different stages of the cell cycle in using mimosine to obtain cells in the G1 phase and nocodazole to fix the cells in MG2 phase. here are some bright field images, and to check if the protocol was done correctly, some analysis in flow cytometry was done, and then in digital holographic microscopy we acquired some images, segmented them, and two studies were done for cells with same subline in different cell cycle phases for cells with great with high malignancy potential F10 in different cell cycle NOC and MIM. You can see that this parameter, dry mass density, differentiate them in very good statistical results. Also, the histograms made with phase shift levels present the same behavior with one peak in for cells in MIM stage and with bimodal histogram and white histogram for cells in NOC stage. Also, the shapes are different, here long shapes and here round shapes. To differentiate cells also from the point of view of malignity level and from cell cycle stage, the same parameters were computed. Dry mass, projected area, and dry mass density, and we can see that these parameters cannot identify different statistical significant difference between the cells with different malignity level when they are in G1S cell cycle. But in G2M cell cycle, these parameters have statistically different values. This is linked also with the biological point of view that cells with great malignity level have high division rate and prepare themselves to mitosis, and the protein content is greater. For the future, we wanted to introduce also some criteria from machine learning to differentiate also these four classes in collaboration with computer science faculty. We tested these classifiers and obtained these results with accuracy over 95% for model using this classifier and shape parameters as features. We search from three kind of parameters: statistic, texture, and shape parameters. In the future, we wanted to expand this data set with parameters of the cell's nuclei. To speak about the fast processes, we performed two studies to see the behavior of cells after electroporation. Electroporation is a technique used already in medical clinic, and for treatment of cancer. And here is some data from the literature. And the medical doctors need deeper understanding about the membrane and its behavior during and after electroporation to elucidate some mechanism about its permeabilization and its repair after electroporation. Electroporation is also used in other methods, not only for cancer treatment. Our first study was made with slow image acquisition. This is the chamber, experimental chamber, electrodes, and here on the bottom are culture cells. The laser pass through these electrodes, and we recorded some holograms before the pulse application. Here is the pulse structure. 4 pulses of this kind, bipolar pulses, positive and negative, and the voltage was and the intensity field was in these ranges. So the laser beam passes through the electrodes, and we recorded holograms before pulses and after pulses from 1 minute to 1 minute. Before pulses, we change the liquids to compute also from the decoupling procedure the refractive index and the height of the cells separately. And after the pulse application, we monitor the cells from 1 minute and also change the liquids after 10 minutes to see again the values separated for the height and the refractive index. Our analysis showed that the refractive index have a great decrease in the first moment, and then it recovers the value from control. And here are some graph representation of the behavior of the cells in time, in all 10 minutes from the projected area, optical path difference, and dry mass. We observed that no changes in dry mass, but in optical path shift there was changes, and they explained that through the circulation of the liquids through the membrane. And one recent study published last year was also on electroporation and fast image acquisition. We used the Photon CMOS camera and recorded 5,400 frames. Each cell is a movie, and 100 frames are before the pulse application, and then 1 minute and a half we monitor the cell, and there also use the same pulses like in the previous here, the same pulses, and the electric field was also in these ranges. And at 1 kilovolt per centimeter square, we have some cells which has irreversible electroporation. The membrane swell very much. Here, these images are also quantitative phase images reconstructed from the series of holograms recorded in the experimental setup. We computed parameters, global parameters as projected area, dry mass density and energy, and also computed some local parameters. We monitored the 3 points along a field line, the highest pixel and 2 other pixel near from the membrane here. Okay, and we computed the autocorrelation functions between in time, and we can see that points situated at very near to the membrane has points have largest different correlation function. In conclusion, I hope that I convinced that digital holographic microscopy has multiple capabilities to monitor cells at a single cell level, cells in attached to substrates, and also digital holographic microscopy is a fast technique to monitor processes. I want to thank you also to colleagues from medicine faculty from Bucharest and my colleagues from Polytechnica University, and thank you for your attention.
Okay, well, we have time maybe for a quick question. Yeah.
Thank you for the interesting talk. You mentioned a couple of times that you use fractals for as a parameter, fractals.
What?
Fractals.
Fractals.
Oh, fractals.
Can you give more?
Fractals,
which kind of parameters are you using?
in this study for cervical cells here, and fractal descriptors here our code fill the surface here and the volume with spheres with different radius, and the numbers of these spheres we computed. This is the fractal dimension. Fractal dimension represented in a graphical representation with logarithm from number and logarithm from radius gives us linear dependence with slope different for small radius and for great radius. And then we computed the mean and standard deviation for these slopes and we made the Gaussian representation assuming that they have a Gaussian representation, and from this representation in the literature the Bhattacharya distance is suitable to see the distance between two categories. And we split our samples in these four categories. The fractal dimension is only this ratio.
You have one for 3D and one for 2D. Two dimensions.
For 3D in volume, 2D, this is in surface, excuse me, here is 3D in volume, and here is the surface, but also the surface is...
It looks like the surface fractal dimension is bigger than 2 on the next slide.
Where?
So, for example, the first one, first distribution, they're above 2. Is that possible if you're in 2D to have a fractal dimension that's bigger than 2?
I don't know.
Oh. Okay, I think in the interest of time, we need to thank our speaker one more time. Thank you. Moving on to the next talk by Bjorn Kemper, who's coming from University of Munster, and he's going to talk about QPI using digital holography for also cell dynamics. 2 words in vivo. That's hard.
Yeah, so let me explain the title a little bit. So, because I will not present any new experiments here, so I will just become just very close to the patients, and so I thought it was a very good title to have it, just to avoid misunderstandings. So first of all, thank you very much for the invitation that I can be here and that I can present what we are doing in Münster. And yeah, maybe it's good to know where we are from. So we are located here in the west of Germany, and we are part of the medical faculty, which maybe is not unique, typical for such kind of technology development. And another thing with regard to the presentations yesterday on the nanoparticles, we are included in the nano bioanalytics center in Münster, and we develop on one hand optical technologies, but on the other hand also we characterize biological effects that come from nanoparticles, toxicity effects. And also good to know maybe that we are having several label-free technology we are using. One is QPI with DHM that we develop ourselves, but we also have commercial systems for tomographic phase microscopy and optical coherence tomography we are routinely using for many of our projects. And we're doing a little bit hyperspectral imaging and imaging processing, of course, as well. So the outline of my talk is first I will show how we do digital holographic microscopy. So thanks to the previous talk, I can skip many things. So I'm very glad for that, also because then I can focus on different things. And then I will show some extraction of biophysical parameters, maybe also which have not so much mentioned up to now. And then I will show 3 application examples. One is nanotoxicity testing. The second one is quantification of inflammation in ex vivo tissue. And finally, we follow the leukocyte alternation in a peri-operative course after cardiac surgery, so which is quite close really to the patient. So let me start with digital holographic microscopy. So of course this already has been mentioned several times, so just I make it very short. The main thing for us is that we can measure label-free. Also that we can measure quite fast, and this allows us also to operate in a non-vibration isolated environment. and that we have a lot of numerical methods for image correction, and in particular subsequent numerical refocusing is very important. And also we have interferometric sensitivity, so this is what we reach mainly, and we are limited by the diffraction limit, so by normal microscope objectives. So we are using mainly the quantum phase imaging feature in our techniques. We have also seen in the last presentation many applications in the biomedical field, and also yesterday there were many examples presented, so I skipped this here, but it's also important to mention that the holography is very famous by Lindsay Tech, I think, for technical applications, especially for very fast information analysis, but also I think Gabriel Popescu has presented also something like monitoring of etching processes, or there are also photo disruption processes that can be very fast monitored. So in our lab we are focusing in particular to use DHM for in biomedical laboratories. So this means we have done several, mainly engineering efforts to integrate systems, like as shown some of our recent prototypes here, that we just have very few cables. We can combine it with commercial stage top incubation chambers and so on. We have used a fiber optical system that we are using off-axis holography because this is quite flexible here for integration and combination with various microscope lenses and then all optical and also lifestyle imaging components. We record hologram from which we calculate the face image, and we also can do correlative imaging with white light or perhaps if necessary fluorescence microscopy. So as already mentioned in the previous talks, so the main important thing is the phase shift we are measuring. And what I would like to point out is that we normally have only the cell thickness, for example, and the refractive index, one equation, two unknowns. So we need some more information if we want to really determine something else, the other one. But there are certain possibilities to do it. And over the past years we have developed also several reconstruction algorithms that we meanwhile use routinely also for lifestyle imaging experiments. So these are some systems that we have in Münster. So these three are in our lab. So this is one thing which is a big plastics incubator with much space for operation and to do many things. So this is something we use now for automated measurements, and which can be used with 96 well plates, and also for some certain applications we can change the wavelengths with some supercontinuous sources and a special micro-interferometer system from blue to the near infrared range. And here's also another system we created for the Institute of Hygiene. There was also in previous talks an image shown from the experiments with this institute. So we have here an S2 level laboratory where it's also possible to measure on more serious pathogens. And we developed an integrated software over the past years for several purposes. So we are mainly measuring on three different types of biological specimens. This is on one hand suspended cells, which is quite easy to analyze because they are spherical. We do dynamic measurements on adherent cells, and as well as tissue slices for example, tissue quantification, the density. So some biophysical parameters that we have found quite useful. I want to illustrate this also by some selected application examples. So on one hand, here is a warm heating experiment where you do a stretch and then afterwards monitor the cells migrating into these cell-free area. So we're monitoring also the area, the dry mass we can monitor from segmented images, and also if we have refractive index information, we can also monitor this over time. And this depends, of course, on the cells you're using. Here it works very well, so you can really nicely segment the cells. You can see that there is different slopes of area and dry mass, so you have additional information from the 3D information you get by the DHM, and also you can measure maybe the thickness of the migrating cells into the gap. So another thing is we have developed small tools for cell tracking, so it's very easy because when you just monitor the maximum height, you can do it very computational efficiently and also quite reliable. But of course, it's not interesting for only to have 5 cells in the field of view. You need more, so you need to automate it. And from these trajectories you get, you can subsequently calculate various parameters like mean squared displacement, directness, velocity, and so on. This depends, of course, on what is of interest for the experiment. So you need more, so this means also we need some automatization process. For example, to get something like here shown for bright field images, where you see cells migrating in the field of view. So you need many cells to be monitored, and because it's easier to track the cells in quantitative phase contrast, we can do this here almost simultaneously. And you see here some black spots that show also the dividing cells. It's maybe also an idea to count it. Up to now we did not do it. And then you can just almost automate it with some manually selection afterwards, of course, get 3D trajectories, and afterwards you transform it maybe to some kind of spider diagrams. This is shown here for 4 different pancreatic tumor cell types. You can see immediately the different migration patterns here, which are somehow related to the metastatic potential, and you can do also statistics for several parameters to find out what is important or not, and this works quite well. So another thing is if you fit a sphere to cells in suspension, when it is then it is possible to decouple the radius or the cell volume from the refractive index, and we did it in the past for several different cell types. You can see here, when you sort by refractive index, you get here different volume signatures and dry mass signatures, so you have individual possibilities to distinguish between cells to a certain extent. And sometimes, as here shown for these pancreatic tumor cells, we can even distinguish them. However, this is not always the case. And what is also interesting, or that it is really possible to measure these values several times when you would take the cells out of or at different passages, for example, at different days or months. You get always the same values, which significantly almost do not differ for volume and dry mass, and so you can use it maybe also for cell culture quality control. So, so now the first topic of our recent results is nanotoxicity testing. And first maybe an overview why and how we are doing this. So we are involved in several projects for nanotoxicity testings, and also for which consider regulatory aspects. And therefore we test, for example, such kind of nanoparticles with relatively low toxic potential. This can be particles that are used for biomedical imaging, for example, or something that can be used as drug carriers like these PACA ones. And on the other hand, of course, you need some positive control. This can be digitonin, as my chemical knows, is the membrane verbalizer, and then we need also cell culture controls. So this means we need to do a lot of experiments and also a lot of replicates, and so this is why we can do it now in 69 well plates. And also, of course, we are doing validating experiments with viability assays, and here with WST, for example, is polyometry. So why is DHM here of interest? On one hand, we have yesterday seen that many nanoparticles absorb, so this means we have potential interference with some polyometric assays that are based on absorbance, or that they are based on fluorescence. So here, because it's interferometry, they are only very weak dependency. Of course, if you have high nanomaterial concentrations, then you can have some scattering. But the other thing is, if you don't know what the particles do, you do not need previously defined endpoint. So it's a new technique, maybe sometimes time-consuming and also expensive. And so here you just put it into the system, and then you just monitor until it's necessary, and then you can stop. And afterwards, we decided, so to use something like a dry mass increase after increment after 24 hours to find out and to visualize differences and to determine statistical significances. So another thing is it's also possible, but up to now we did not do it. So we have also, if we are interested in exactly to work afterwards with these cells, with other analysis, of course we can do it afterwards, detach the cells and put it to another chemical assay. So you see also we need automated measurements, and typically we are using at the moment 3 replicates. We measure 12 fields of view for each particle and concentration and so on. And so this creates a quite huge amount of data. So another thing, what I have not talked about here, we also use different cell types in the same experiment, because here we have 4-cell matrix which are from different organs. This means we have macrophages, so it's the immune system, we have fibroblasts, connective tissue, we have kidney cells, and we have lung epithelial cells, and all these have different morphological features. You can see, hopefully, in these very tiny images. And you have also different responses, of course, on the different nanoparticles. So what you see also here, we have quite weak contrast, so this is why we decided here only to evaluate the whole field of view. So because it was not possible to do image segmentation here at the moment, but maybe in the future it's possible perhaps with AI-aided segmentation algorithms. So at the moment we're doing some tests and they are quite promising. So then we get many curves, you can see. So I won't go into every detail here, but you see here the different cell lines, you see here the different particles. And what is also very remarkable here, you can see the orange curves that shows the control for each experiment, and you see we have also high reproducibility of the results we can achieve with our systems. So the main conclusion is here, so we can measure the effects and quantify the effects here in this validation experiments, that we are in good agreement with the WST8 viability assay, but also we have there's some differences, because the one is chemical and the other one is biophysical. The second example I would like to show is quantification of inflammation in polar tissue. And here this is, let's say now a story since a few years, where we started a collaboration with the Department of Internal Medicine B at the University of Münster. And the motivation is that we have inflammatory bowel diseases like colitis and Crohn's disease. There exist up to now not really good markers to identify if a patient has reacted positively on the treatment, or to really good to identify the degree of the inflammation. And therefore, we tried first in an animal study with a mouse that were treated with a substance that they developed inflammation of the intestine. So we monitored afterwards parts of the colon. Here, this is healthy. This is swollen, inflamed tissue. Of course, you can see it here by eye. But it's very easy here because by tissue sections you know the thickness, and so you just can transform these quantitative phase images into density maps. And so you can see here the differences visualized by cross-section, and you see that we have for all mice with colitis, we have a significant decrease of the refractive index in these areas, in all these tissue areas. And after these promising results, we also tried the same with patients. So here we compared patients, biopsies from patients in transmission, in remission, together with an active Crohn's disease event, and then we got the same behavior. So we also have similar results. So in this case, it's quite promising approach to quantify here, for example, the degree, and also what I show not here, but it's shown in one of the papers here, that it's also well correlates with other clinical parameters like the body weight of the mice. Then also after low inflammation, we have also some stenotic effects, and there in the next experiments we investigated what can we measure for stenosis. And here you can see, we take also bright field holograms and calculate quantitative phase images from that. What we can see here also, but in difference, we have, for example, and from our validating experiments, we think this is collagen, which is going inside, and we can see here also a slight density change, and here we have the opposite effect. This means we have a higher refractive index in these stenotic areas than in the almost normal tissue. And this was also verified here by a pilot experiment with AFM-like system, where we had some support from a company in the Netherlands. And we also found that we have here a higher Young's modulus. So we also can relate a reflective index here somehow with tissue stiffness. And we did some correlative investigations with immune fluorescence markers, fibrosis markers. And here we focused on collagen 1, 5, and telastin. And so we also get a quite good correlation with these parameters. So I think this is a good, promising result, and at the moment we are currently further increase our data with further measurements as currently done by a PhD student from the medical department. So the last example I would like to show is that we tried in a pilot study to follow leukocyte alternations associated with perioperative inflammation. And here after cardiac surgery, we had a collaboration with the anesthesia department. And here's just the overall concept of the study. So before, two hours before the operation, and then at day 1, 3, and 6, we took blood from the patients. The sample was divided. On one hand, we separated lymphocytes and monocytes for DHM investigations and analyzed dry mass, refractive index, and form factor. So, or it's also circularity of the cells. And the other part was then analyzed with common inflammation-related markers by flow cytometry, and then we correlated this and found out what can we get out of this and what we can conclude. So here just once more an overview about the study. So here we because it was a pilot study. A PhD student from the medical department really per hand selected all these 150 cells per patient, and this was done at day 1, day 6, day 3, day 6, and also he did everything from fresh taken blood all at the same day. So one measurement was was from 7 in the morning and in the evening at 9 o'clock, and then we had got all the data, and so what we see here from refractive index and size data of the cells, that we can really nicely already separate between monocytes and lymphocytes, so at least much better than in the flow cytometry, and this is for almost all patients the case here. But when we look at the whole collective of all measurements, and so we have here now 30,000 cells, really everyone manually evaluated. So we can see here some kind of trend. So we show it here as a movie, and we have some cells scattering in one direction, especially the volume has been increased. And if it's difficult to follow, maybe you can look at these regions of interest where we have marked where the cells scatter. And we can also see this trend for the individual patients. So we have some increase of the volume of the monocytes in particular that decrease until day 6, so getting back to normal. And we also have here some correlated refractive index changes. So however, we do not, at that point, not really know what is this at all related to, because we just wanted to see if we can measure something. We had some promising publications found concerning sepsis, so this was the starting point. And what we did now, we correlated all our parameters, flow cytometric, laboratory parameters in a bivariate correlation, what changed from day 1 to day 6 to day 1 with respect to the before the operation, and also we tried the same at day 6. And so this was a quite huge analysis where we had a statistics department involved. And then we found that we had several correlations, so it's not a very heavy correlation, but it was found significant. And we found all what we found was quite plausible, so we have several markers concerning lymphocytes and activations and so on, and this all was related to volume and refractive index changes, and also we found some small form factor changes to some inflammatory marker, which may be something related to surface change of the cells, perhaps. So this is so we regard this now as a basis information for further, more focused studies. But we also did another thing, that we compared now the DHM data with data concerning, on one hand, the course of the course, if it was regular or if it was complicated, or if the patient received epinephrine, so for stabilization, and also the CRP inflammatory marker. And we found here some similar developments and completely different development. And afterwards, this was also upon an advice on the reviewer. So we found here that this really very well reflects our patients, because there was no correlation here between the CRP level and the epinephrine, so you can see here, but we have a really good correlation of 6 patients with the serious course. And also we investigated if this is also not only for average values, but also in the temporal course. It's maybe not so easy to recognize, but we have a trend really that almost all patients have the same trend with the parameters in perioperative course, because But you also, on the other hand, see, because it's a limited number of patients, it was 25, so we also have a lot of huge variations, and even in the size of the blood cells, and the refractive index, which is really individual for each patient. So nevertheless, we found several good, interesting correlations, and so we think this is a good basis to go further and more focused to characterization of inflammatory processes and with respect to the course of the disease. So already I'm at the end of my presentation. So quantum phase imaging with DHM allows so on one hand quite nicely leads to nanotoxicity assessment of organic nanomaterials. Here we found that dry mass development dynamics implement were most interesting. The quantification of inflammation tissue was quite easy to use the refractive index here. And with this preload knowledge, we also found that inflammatory processes in blood cells after surgery here with inflammation-related processes can be monitored by form factor, refractive index, and cell wall volume. So one advantage is really that we have absolute parameters. It allows better comparability. We found many correlations really with other chemical assays and clinical parameters. And future directions, you see, automatization and simplification is very important. Otherwise, you cannot do all these experiments because you need to acquire a huge amount of data and to analyze it. The second thing is, of course, which has been addressed yesterday, also more enhanced specificity. But also, it's important to have comparability if other researchers want to repeat results. Although we measure optical path length changes, the whole experiment needs some degree of standardization, in particular for toxicity measurements. So, and finally, we need robust laboratory systems, simplified handling, and for example, machine learning AI-assisted analysis for evaluation. So, and finally, of course, you can only do it if you have a lot of collaborations, fundings, and company cooperations. Thank you very much for your time.
Any questions, please?
Thank you for this presentation. It's very nice. I'm not so familiar with this, and I have a short question. If during the measurements you can add algorithms to see the cell signal in the, I don't know, or you have to incubate the cells before the starting the procedure in algorithms?
You can use it, for example, in a flow chamber where you exchange the medium. This is possible.
Yes.
So
during the
this is possible.
Thank you.
We have some small fluidic chambers and so on. This would be possible.
Thank you very much for a very interesting talk. I have seen that you have shown a tissue in a couple of cases from disease and sterility tissue. And I know that the thickness in both cases was 7 microns. I would like to know if this was a compromise just to have a scattering or whatever.
In fact,
or is something that doctor give you the sample that side?
No, so indeed we made some experiments, but also we wanted to meet a compromise that is a normal thickness that can be also used for HV staining and so on. And so it works with DHM, but it depends on the wavelength. So if you switch to infrared, you can go beyond a little bit 10, and so it's a normal thickness. But if you would try to use deparaffinized tissue, on one hand, of course, you have to consider refractive index, it's not easy. But then we found 3 microns is the best. So of course, this needs some trial and error, and but also you have to keep in mind that it should be compatible with common procedures in pathology.
So a lot of the applications that I've seen from you and the other speakers typically try to match what you see with this methodology with other methodologies that have been used. There are labels, fluorescence, chemical. Do you find situations in which you can actually, you provide them information, so you identify a biological process by using this methodology that they cannot?
This is, I think there are several, but it's difficult to explain, of course, at the moment. So, and this is so, firstly, we try first to understand what we see, and then we can differentiate what we do, what we not see, because it's very difficult to convince without sufficient knowledge. But what is, for example, something in the preparation thing also, we have several different fixing methods, for example, for HE staining and so on, and these significantly you can change the morphology of the tissue and so on. And here's the QPI, something that can be very precisely measure these changes in comparison to fresh or to cryosections or something like that. So this is something we already have done, but then we have to see if people would like to know it.
So this is, I can add to this, this is clearly a question coming from a clinician. Because in the beginning, like Bjorn showed, we also got images that kind of recovered everything from the H&E. And the doctor said, we don't care, H&E is great. It's very simple. Can you give me something that I cannot do with H&E? But Bjorn and also our lab showed that actually collagen is something that doesn't show very well in H&E.
It's extracted or
So, but it shows very well in phase. So actually, that's what got them excited. So now you can use collagen, as you showed, for certain diseases, also for prognosis in cancer. So that is actually a good question. So just recovering a standard procedure that already works doesn't get them excited. It's what you can add that's not there. So with that, thank you very much, Bjorn. Next talk comes from our local host. Mihaela Gheorghiu is going to talk about high content dynamic imaging of single cells.
So it's really nice to be here, act as a host for this interdisciplinary conference. It's a pleasure for me to give this lecture, but it's also a very big challenge. The challenge can be seen directly from the title, because in a way it just emphasizes several aspects which were instrumental to this meeting. So, I'm talking about high content, label-free analysis, dynamics, single cells, and also about something which is very new to what was discussed currently. It's the electrical information in a modulated microscope, let's say. It's also very challenging, this presentation for me, because this amount of complexity could not have been possible without support of a wonderful team coming from Bucharest, from the International Center of Biodynamics, and also some nice inputs from Gabriel Popescu's group, Sorin Melinda. I was hoping to have him here in the auditorium. Claudiu Supuran, who will probably join us online, and Tudor Bada in a very special application. So I would like to start my talk by acknowledging their contribution and providing rather atypical thanks at the beginning. Of course, it would be nice to also thank the funding of what I'm gonna talk about. The third challenge in what concerns my talk is that what I'm going to present are very, very new results, and well, I hope you will join me in showing the same excitement as I will probably show. So, why high content label-free assessment of single cell dynamics, and why based on electrically modulated microscopy assay will hopefully be revealed during my talk. Just about cell dynamics, it is known that cells which are in suspension at the beginning, they are adhering to the substrate in processes which have a different dynamics. Then they decide to proliferate, differentiate, die in a cell and context-specific, again, time scale. And these early events in cell signaling are having the second time scale, while intracellular signaling processes and transcriptional changes can take minutes or hours. And by contrast, cell fate decision can take many, many hours. So being able to span all these time scales is quintessential in monitoring cell dynamics. So there comes the next question, assessment of cell dynamics. What's the good of it? Of course, here in the Center of Biodynamics, we are very much interested in developing cellular platforms and using cells as biosensors. And for that, it would be also probably interesting to show that we are able to devise cellular sensing platforms that reveal enhanced sensitivity when modulating cell homeostasis. It's also a continuous process of development. We used cellular sensing in order to, for instance, reveal diverse processes in the effect of amyloid fibrils involved in Alzheimer's disease on living cells, or the quantitative assessment of carbonic anhydrases inhibitors when using hypoxic cells. So what do we need for that? We need combined measurement methods and related setups to gauge biointerfaces and cell signals, which would be ideally label-free, non-invasive, and multi-parametric. Like all the tales, because the symposium is having this tail dimension, I'm starting with once upon a time, because electrical cell substrate impedance sensing is a rather classical approach in our center. But what we are interested in is to gain detailed information concerning the key processes typically revealed by empirical measurements, which are cell substrate adherence. We saw in previous talk about cell spreading and attachment, and derive the information regarding cell state, cell response to various cues from the substrate. To see variation in cell shape and volume, I was intrigued by the new term introduced, morpholomics. So yes, being able to derive morphology information, but also more structural, more functional information, like the efficiency, how functional the cell-cell junctions are, and see how cell-to-cell communication is taking place. For that, typically impedance is recording or varying the frequency of the applied field, and also uses time evolution to derive information concerning special structures within our system. So Why high content label-free assessment of cells? We now know, are very much familiar with this image, and we want to add information concerning cell-substrate interface, which is provided nicely by reflected light contrast. Being able to derive information on intracellular structures which are typically seen with fluorescence, and using microscopy assay, being able to derive electrical parameters that are revealing cell-substrate interface and cell-cell communication. This is also classical view of cells, in which we want to see nucleus, but also these intricate structures, the ruffles and the cell cortex, philopodia, and hopefully the intracellular structures related to energy production or lysosomes, for instance. So we move to designing an electrically modulated microscopy assay able to provide high content label-free assessment of single cell dynamics. The purpose is to achieve time series of maps of the impedance and optical parameters of bioelectrified interfaces with optical resolution using optical means. Clearly, that would mean a quantum leap that would bridge scanning electrochemical probe microscopy and microelectrode arrays information, which rely unfortunately on extensive nano microfabrication, have reduced time resolution, limited signal to noise ratio, and increased cost, and also require integration with complementary techniques for validation. Electro-optical imaging is not new in the literature. I'm not talking about fluorescent markers for the membrane potential, but I'm talking about non-fluorescent optical reporters exploiting the intrinsic refractive index and surface charge density, or voltage sensitivity of noble metals, as we saw yesterday in Professor Ashtilan's talk. Or thin films, like in plasmonic electrochemical impedance spectroscopy or nanoantennas. One of the pioneers in the field, which I cannot oversee, was also mentioned yesterday, the group of Professor Tao, who unfortunately is no longer among us, that showed that one is able to resolve subcellular structures and processes without labels. So our goal is to have high spatial resolution and low temporal noise multifunctional imaging instrument capable to perform plasmonic free impedance mapping. And this should be done only via microscopy data. The initial or the grounds for this, the proof of concept was shown recently with an article showing high resolution impedance mapping using electrically activated quantitative phase imaging. And we also introduced there the concept of reflected light coupled with phase imaging and electrical information derivation. So we started with raw images of refractive index contrast. We derive nicely pioneered by Professor Popescu's group and provide individual epi fluorescent mist images from which we derive phase, and when applying an electrical modulation, we can also perform Fourier analysis and show the amplitude per pixel for the applied frequency. This enables us to derive simultaneously the phase information and also the electrical map of the sample. And the sample at the time, it was a nano-engineered substrate, and shows that indeed we are able to achieve phase contrast and electrical contrast on the surface. The images show with high intensity the conductive areas, and with low intensity values the high impedance areas, dielectric surfaces. And it's interesting to see that one is able to get pretty good signal to noise information, only visible in the amplitude of the signal. What is really important is that one can derive the different information concerning the heterogeneity on the substrate, and also compare the electrical information with the standard data. We were interested to apply the same approach to transmission data, and well, to a certain extent, we were able to map both the impedance substrate and the phase of the cell which was adhering on it. We were also capable to use high spatial resolution electrochemical biosensing to demonstrate high spatial resolution electrochemical biosensing using reflected light microscopy on itself. And that was very nicely shown by Sylvester Gaspar and our team. The capability to follow electrochemical signals from electrodes which were modified with certain enzymes. So we decided to use reflected light microscopy into analyzing both substrate and cells adhering on the substrate. And we rely on the constructive and destructive interferences of the reflected rays generated from the incident light hitting the interface with different structures within the cell. And we are happy to see that we are able to differentiate the conductive and non-conductive areas and provide information concerning the electrical contrast of both the substrate and the cells adhering to the substrate. We moved further and we managed to increase very much the amplitude of the signal, and that enabled us to derive actual impedance maps of the cells. So how do we do that, and what is the relevance of this type of approach? If you compare the DIC, the differential interference contrast images, with the electrical information provided by bright field reflected light and the original bright field reflected light, one can see that we are able to not only see structures which are related to the nucleus, but also to see areas where cell-cell communication was able to take place. Just have in mind that these are fixed cells. And it's interesting to note that features which are seen in bright field, electrically modulated bright field images cannot be extracted from neither the DIC or the bright field, original bright field data. It is interesting to note that the capability to reveal quantitatively and dynamically and assess the dynamic distribution of intracellular structure and electrical parameters with sub-micrometer resolution is provided primarily by this coupled AS and optical microscopy. So now we are moving further to live cells, and it is interesting to see on this very well peculiar model cell that indeed electrically this is an image which is derived by electrical modulation. We not only see the typical information like the cell cortex, lamellopodia, or roughly, if you see how the shape of the cell is developing here, but and also we can see that the cellular cortex is visible as these well increasing complexity structures. We also see that there are definite areas in which charged particles are generated inside the cell, and also we can see some actin cables, and the cell junction is an area with active electrical information. So that brings me to the first impact of the data, which is the fact that one can gain functional information concerning the cell-cell communication. And while this is definitely a very large complementarity between the optical and the electrical information, this shows that this is an area in which we can definitely collaborate. It's also possible to see the nucleus inside the cell. So I liked very much in the presentation of Nathan, his sperm cell having glasses. So so I couldn't I couldn't refrain myself from showing what is rather a cellular zoo, which is visible in the electrical information, and that brings me to what I consider a very big challenge in the morphometry, which was introduced. So despite the fact that these are the same type of cells, one can easily imagine that this is Brancusius bird, so Myastra. This could be seen as whales, at least for me it was definite, and this could be seen as stingrays on the surface. So what is the impact of it? I don't know, we have to discover them soon. But it's also interesting to note that those areas are definite regions in which signaling with charged particles is taking place. What we can do is we can derive the same impedance spectroscopy data as shown for the nano-engineered substrates, and derive from here either typical histograms, but also see the frequency dependency of the mean intensity, which is proportional to the impedance information, but also a rather peculiar data, which is the skewness, that shows how many pixels are lighter, so how much symmetrical is the histogram. So it's interesting. Those are data from 2 cells, and the skewness shows the same evolution for the frequency dependence. While there is something that should be investigated in terms of the mean intensity of the whole image. So what we can do is we can derive impedance map, which are now only seen as individual pixel intensities, having in mind that low intensity means low amplitude of the Fourier transform, which is related to low surface charge density and a higher impedance. So the impact is that we can show optically derived impedance spectra on live cells. So this can also be quantitated during the exposure to a certain stressor. Now I'm showing how cells are behaving on the surface upon 1 hour and 30 minutes of exposure to a compound which determines a certain membrane permeation. We'll go back to this issue, but what is interesting is that one can see that the cells are really retracting from the surface, and also that some legs which are belonging to the lipid membrane occur. So the fourth impact would be that the optically derived impedance spectra can be used to assess stimulus-induced cell dynamics. So I'm grateful for the presentation of yesterday, in which colistin was shown as an antibiotic of the last resort. The question is, last resort means that, well, the side effects of it seen at the level of even neural and also some gastric problems are prohibiting their use. So we choose to use colistin on mammalian cells to see how they are behaving. And the colistin level is similar with the one which is considered the minimal inhibitory concentration for bacteria. And what is interesting to see is that after 30 minutes of exposure, the cells are really reacting to the presence of the colistin. So impact number 5 would be label-free assessment of antibiotic effect, this time directly on the cells, on the mammalian cells, and see whether this last resort antibiotic, it can be really used in well, not only immunocompromised patients, but probably also patients with neuronal problems. So what we see is that the membrane is permeabilized, there's cell retraction, but also extracellular vesicle formation. So the next question was, is it possible to assess cell viability or cell vitality? So we turn to the what is known as a standard viability test is the MTT test, and we try to see whether it's possible to assess the effect of colistin together with the MTT approach. So yeah, this is the DIC image which shows not only color change in the outer surface of the cell pair, but it's also visible that things are changing inside the cell upon MTT exposure. So I would like to draw your attention to the poster, which will show some data on MTT and the production of formazan in relation to live cells. So what we see is indeed membrane permeabilization, cell retraction, and intracellular granule formation. The electrical information shows that the granules appearing inside the cell are electrically active. It also shows that what is happening at the level of the nucleus is corresponding to a decrease membrane impact, so there's clearly a permeabilization as well. Also, in time, electrically, what one can see is that cells are totally retracting from the surface, and the membrane is completely permeabilized. So the relevance of it would be that we can get label-free single-cell vitality assessment, and it's also important to note that we can follow the steps involved in enzymatic production of formazan in the entity, classical entity approach. We see the entity uptake, the reduction, which is detected by appearance of the staining in the cytoplasm, and the compartmentalization, which is the appearance of granules and the clearance of spread staining. And we were also interested in deriving the formazan extrusion and the appearance of needle-like aggregates on the cell surface with the clearance of cytoplasmic granules. It's interesting to see that indeed these needle-like fibers are visible in the electrical information, and we also see complex structures like threads woven of the thinner fibers extending throughout the cytoplasm. We also show that these thin protofibers are the ones that determine membrane damage with formazan granules and aggregates forming rapidly after the membrane collapse. So the impact is clearly the ability to monitor formazan crystal formation and derive the viability at the individual scale. And something that we are very much interested to explore is the relevance of this data for virus infectivity. Just as a glimpse, one can see that the formation of syncytia when one cell is infected would determine the modification of cell-cell contacts. And we currently run a project concerning high resolution and multivariate dynamics at single cell level for viral detection and assessment of cellular response to this exposure. Another relevance is the ability to assess the bacterial cell dynamics and perform the antimicrobial susceptibility testing. And it's interesting to note that we can complement the information on transmitted light or DIC with the reflected light information and the electrical one, and reveal also this kind of shadows which are corresponding to cells actively dividing on the surface, and also has an implication for chromophore media, the one that could be used for identification. So we can also, foresee analysis of biofilm formation and invasion in hydrogel, and also real-time label-free live dead discrimination of bacteria. So please also have a look on the poster concerning quantitative evaluation of bacterial cell dynamics in managing disease. So, of course, this doesn't come without challenges. The technological one is the ability to achieve optimal light intensity, frequency range, and computational resource for optimal electrical feature extraction. It's computational wise to achieve validated multi-parametric assessment. The question that arises, what is the effective training library for implementing, for instance, artificial intelligence augmented analysis? Biologically wise, it's the selection of relevant cellular models and the availability of complementary information, and concerning material sciences to design optimal surfaces and cellular platforms. So instead of conclusion, I would like to pinpoint some future perspectives which are opened by the capability to achieve multi-parametric label-free phenotypic features of cellular-based sensors. We're talking about achievement of label-free dynamic information on cell components at various levels, and importantly of electrical maps, which are able to show cell substrate adherence, cell-cell communication, and membrane integrity. Achieve multi-parametric label-free sensing platform for quantification of viral particles, and also achieve faster and more relevant platforms for antimicrobial susceptibility testing. Of course, this was not shown here, but it's a dream come true to be able to monitor the electrical propagation in neurons. So this image, you know, from yesterday, but I like to highlight the fact that this is the old and this is the new, the bridge just across the river. So I hope that this, not only my presentation, but this whole meeting will be able to show the nice future ahead. So with that, I thank you for your attention.
Thank our speaker.
Okay, so very nice presentation. I have 2 remarks and 2 questions. Let's start by the remarks. First, I think that this is really nice because it gives us new label-free data for deep learning. It's not only handcrafted feature, it's deep learning. You can give these images directly to classifying networks, and then you can get, you can correlate it to pathologic states. Now, second remark, if these are quantitative maps, it's better to add the color bar with the values right next to the map. Now, these were the remarks. Now, the questions were, what are the challenges if we want to integrate like this type of measurement method with QPI? I can think about 2 challenges. First, what is the challenge related to the substrate on which you put the cells on? Because we have our own limitations when we image it. It needs to be transparent, it needs to be filled with the liquid in there. And the second one is, would you think that there might be interaction between your measurement and our measurement by combining them? For example, will your measurement affect the refractive index or not?
So the response is like everything else, multivariate. So thank you first for the comments. Indeed, I look forward for interaction on these artificial intelligence approaches towards our data, because I feel that there's very much to learn and to extract out of them. In particular, since they are very new information-wise. The second, I don't to remember that.
No, that's the color bar.
Okay, okay, the color bar was given only for one image. And again, it was given in intensity values and not in impedance, because one has to have a certain calibration, initial calibration in order to, I don't know, add real values, impedance values on these. So now coming to the question. We are very much interested in combining the phase information on our data. Technically wise, there shouldn't be any issue with that. If we are considering very thin chambers for your application to work, I see some impact on the particles which are outside the cells. That has to be somehow cleared. So we can have whatever thickness of the measurement chamber, and the substrates are fully transparent. So,
what about the pathogen that changes due to your measurement?
What is changing is the refractive index of, well, there's a dependency between the surface charge density and the local refractive index.
Right.
And this surface charge density is dependent on the impedance of the surface, and the impedance via the dielectric information is related indeed to refractive indexes. But we are not changing the refractive indexes of the substrate. We only reveal the shadow, the image of the surface charge density on the substrate. So from this point of view, our measurement is not destructive on this type of information, but on the biology of it, we took special precautions to minimize and fully eliminate the impact on the biology of cells, because we are talking about capability to change the wavelength of the light, which is the incident light we are using for bright field reflected light microscope.
How fast can you go? That's the last question.
Very fast, because there's no limitation on depending on the camera you're having and on the frequency you are applying. Yesterday during the talk, it was mentioned that while ideally we can go for a wide frequency range, there is this dependency of electrical information, which is manifesting highly on a rather narrow domain. So I was showing data with the actuation of 2 Hertz, but
Kilohertz would be
kilohertz would be
possible, like images in kilohertz.
Yeah.
Okay.
We didn't show it, but we are very much into using fast cameras.
Very nice.
Are there, please.
I enjoyed your lecture very much. I have a general question. Most of these experiments are using cells, isolated cells. I was wondering what happens with the cell after isolation. The cell membrane is definitely, maybe it's too much to say altered, but anyhow, changes in the cell membrane take place. It doesn't matter the method for isolation. So maybe I missed it, but I was wondering how long time after isolation you can use a cell. How do you know that the cell which you work with is in a physiological shape.
I think these are cell lines. You're thinking of primary cells probably.
Even for cell lines, if you isolate single cells.
So those are cells which are cultivated directly on the surface. So definitely we can also monitor the process of attachment of cells to the substrate. And we use the reflected light shadow, the test of cells adherence point to the substrate. And one information would be to have an even contact of the cell to the substrate. And we kind of guessed it. And we can also enhance the attachment by using lysine or attachment compounds. But also, we managed to have cells to adhere on the substrate rather.
Sure, yeah, I was wondering.
I understand what
How long time after you put the laser cell in the culture, whatever, you use the cell for your determination, or in the other method, any determinations, after culturing it?
We normally have it overnight. So, but this is, I don't know, it's a matter of...
But I'm not sure I understand that the concern is the trypsinization when you put them in
Any method of isolation would do something, I wouldn't say damage, maybe we can see alterations of the cell membrane. So I wonder how long time after isolating the cell or placing the cell in the cell culture, you can use the cell and be sure that it's in.
My experience, they start adhering immediately, and in about a couple of hours, they're fairly flat. But
actually, it depends. It depends on the cells and also how flat they become.
No, it's a general question.
But in general, I think it's accepted that trypsinizing is not that bad because
it is.
It is a little drastic. Yeah. And we are using a lot, so.
Yeah. So,
but did you see reversible changes? I mean, the fact that they go into the cell section and then they're happy dividing.
No, but after trypsinization, then we will use the cells in after several changes, or after 1 week or 3 days or something. That's a general question.
It's overnight.
Overnight, yeah. Hopefully it's okay.
I showed you my personal zoo, and I'm really looking forward to see how they how they are changing depending on the time spent from trypsinization, depending on the compounds, growth media, and so on. So there's it's uncharted territory, as I said, and I think it's a very nice addition to what is classically seen in microelectrode arrays and in electrophysiological therapies.
I think with this, I'd like to thank Mihaela one more time and all our speakers in the session. Are there any announcements, or are we going
You
to...
read my mind as always. So, coffee break, I think that's a must. And as soon as possible, I would like you to join and have another beautiful picture.
Or did I miss one?
I would also
poster session. We have many young presenters and very nervous and I hope
We're my friends, don't worry.
and please let's encourage them, and also for all the presenters, please keep your presentation in around 5 minutes. I will let you know if you go beyond that because we want to keep with the schedule. So our first presenters come from the International Center of Biodynamics. Daniela Tudor, who will present the microscopic evaluation of formulas and products for assessing single-cell viability.
Hello, I'm Daniela. It's very nice to see you all. Thank you for coming. As my colleague said, I'm going to present you a microscopic evaluation of formulas and products for assessing single-cell viability. As you all know, rapid assays and accurate assays for evaluating cell viability and cytotoxicity are highly needed. One of the most widely used is MTT assay, based on the reduction of the yellow tetrazolium salt to purple formazan crystals by metabolically active cells. It's a gold standard, but has many disadvantages, like missing cell resolution, missing single cell resolution, long incubation time, the lack of sensitivity, and the use of toxic solvents like dimethyl sulfoxide and isopropyl alcohol. In this context, we aimed to develop a more rapid and sensitive assay to investigate cell viability and cytotoxicity at a single cell level, based on the biochemic principle of DMTT. Practically, we investigated the formation, the production of formazan crystals within the cell in real time. Our experimental setup, we used as experimental model mammalian cell culture, band 3-cell line. Those are endothelial cells isolated from murine. We cultivated them on button glass culture dishes. We had control and stressed groups. As a stress agent, we used colistin, an antibiotic whose cytotoxicity is well known. We've investigated the formazan crystal production using inverted dark field reflected light microscope. What became new? Instead of adding the stress agent or the test compound and after 24 hours the MTT reagent, diazolyl blue, we added both at the same time, and afterwards, immediately, we've investigated under dark field microscopy. Here I've illustrated some representative videos. Here there are time-lapse images taken in dark field reflected light microscopy with cells treated with diazolyl blue, the MTT reagent we used. Here I've illustrated cells under stress, treated with 2 micrograms per liter colistin. Here are unprocessed images. Here are processed images. We've applied the contrast to highlight the formazan crystals. Here are histograms associated with the light intensity. We saw that the production of formazan crystals is associated with an increasing scattered light intensity, which is highlighted in these histograms. And here there are some drawings obtained after processing, which helped us to assess the crystal's surface or area. All images were processed in software ImageJ.
There is no image.
I was wondering.
This is the control group before processing. This is with the contrast, where we highlighted the formosome crystals, and as you can see, there's an increasing light intensity. Here's the histogram associated with the increasing light intensity in control groups. And in contrast, in cells under stress conditions, stress groups, as you can see, formosome crystals are form much slower compared to control groups. They tend to remain at the stage of simple granule formosome, intracellular located. Here I've illustrated the dynamics of formosome production in time compared to the classic MTT. By using this approach, we can obtain results in less than 1 hour. With blue, I've represented the mean gray value associated with the scattered light intensity for both control and the stress groups. And with red, I've illustrated the area of formosome crystals. So, in conclusion, as I said, our method showed results in less than 1 hour. It allows a single cell analysis in real time. And our results highlight the dynamic differences between cell cultures in normal and stress conditions. There's a clear association between the formosome production and the scattered light intensity, which was higher in stress cultured under normal conditions compared to those cultured in stress conditions. Also, we saw different formosome crystals dynamics in cell under normal and stress conditions, and larger formosome crystals formed by control cells compared to those cultivated under stress conditions. I would like to thank our supporting projects and SAIS for helping us with the experimental setup with the microscope, and thank you.
Thank you very much. If there are any questions, please.
I'm not very familiar with this substance. So the viability, when the thing crystallizes, that's a sign of good viability?
Can you repeat, please?
How is this a reporter on viability? When you see the crystals, that means it's a good thing?
The production of crystals is associated with the metabolic activity of the cells. So only metabolic reactive cells are capable of converting the MTT reagent, thiazolyl blue we use, to those crystals or granules.
Okay, and then are we sure that is not affecting the function of the cell?
It does. It does.
They are.
We're worried about trypsin. Okay. And can you use it as a marker for in the title of that paper that you cited? It was in the lipid droplets. The crystals were localized in the lipid droplets. Can you track the lipid droplets?
Yes, they can be tracked. There's an article regarding this using special stains. They tend to...
Well, I'm thinking to use the crystal as a refractive index marker.
To use the crystal as
A refractive index marker because it's very prominent, and then you can track the liquid droplets, which I think is
Yes.
cool. Okay, thank you.
Thank you. Thank you very much.
Good paper.
Our next speaker is Roberta Banciu from the International Center of Biodynamics.
Wow, that's a language just like
Hi, my name is Roberta and I'm delighted and thankful to be part of the scientific event. We'll talk about photosynthesis. Our work is inspired by an important concern on agricultural field regarding herbicide detection. We employed cyanobacteria thylakoids from Synechococcus elongatus as the biological recognition element due to their stability, high photocurrents, and remarkable sensitivity to herbicides. The photosynthesis process was inhibited by diuron for the thylakoids in solution and immobilized state. First, we characterize the thylakoids absorbed on PDDA pretreated mica using AFM intermittent contact in air. This showed us fused granular membranes displaying various levels of thylakoid stocking, most often with heights multiple of 4 nanometers. Next, the zoom of a single membrane disc confirmed the presence of photosynthetic complexes with diameters around 20 nanometers, which are frequently found in semi-crystalline arrays. For the electrochemical detection, we have to optimize the concentration of chlorophyll. Its value is 5 to 10 micrograms per milliliter for cycles of 1 minute white light and 15 minutes dark. In solution, we used DCBQ as a mediator, so we reached a good sensitivity. The electrochemical signal decreases proportionally to increasing concentration of diuron up to 400 parts per billion. Regarding the immobilization, we compared two types of screen-printed electrode, carbon nanotubes and gold. A drop of the thylakoids, G43 polymer, redox type, and PEG crosslinker matrix was dropped on the surface of the working electrode. And then we have the current, which decreases with diuron. So the best choice for a good sensitivity was the carbon nanotubes. In conclusion, the presence of herbicides was successfully achieved using thylakoids in solution and immobilized, and suitable samples for our biosensors in this current stage of development are environmental waters. In the future, we aim to improve the sensitivity, address the selectivity problem, and employ the thylakoid herbicide sensor in real water samples. Thank you for your attention.
There are no questions. Please, thank you very much. Our next speaker, also from the International Center of Biodynamics. Biodynamics will present the optimized magnetic capture of target bacteria using magnetophoresis and periodic activation.
Good afternoon, thank you for the introduction. I'm first going to start with the idea behind our project, and that is the fact that bacterial contamination and the infections that it may cause are a great threat to human health. Therefore, we are facing a great need for new methods and devices to try to capture, to enable rapid detection of bacteria. So, the aim of this work was to improve the capture of target bacteria. A very effective way of isolating microorganisms from complex samples is the immunomagnetic capture using magnetic particles coated with affinity compounds such as antibodies. So how do we improve the capture? We developed a system that uses magnetophoresis and periodic actuation to shorten the capture time. First, we made theoretical simulations in COMSOL multiphysics. But first I'm going to explain to you how the system works so that you have a more clear picture. So the system operates through magnets that are parallelly placed that actuate periodically upon a measurement chamber. You can see it there. The measurement chamber containing magnetic particles and bacteria. The measurement chamber itself moves so that the magnetic particles and bacteria travel the whole measurement chamber to form conglomerates. We simulated many models in COMSOL, but we chose only a specific configuration of the magnet so that the magnetic field gradient, when the magnet is near the magnetic particle field chamber, is the most powerful. Here we have a particle tracing type of representation of magnetic particles when the magnet is approaching the measurement chamber. So let's imagine this square is the measurement chamber. The magnet used to be here, and it went on the left side. You can see the moving trajectory of the magnetic particles traveling to this side of the wall, and here these are the magnetic particles compiled. For the actual experimental setup, we have, as I said, so this is the measurement chamber, these are the two magnets that actuate upon it. You can see here an even closer picture. And this method, we verified it to take about 10 minutes, and we made a comparison to the classical method, which is the incubation using a mechanical agitator, such as a rotating incubator, for instance, that takes up to an hour. Here's an experimental demonstration. I tried to capture what happens in the measurement chamber, so you can see one of the magnets on this side. These are the magnetic particle cluster. It goes away and the other one comes, and you can see it traveling through the fluid, piling up on this side of the measurement chamber. For validation, we analyzed these conglomerates of magnetic particle and cells in fluorescence microscopy. Here you can see the method with magnetophoresis and this with mechanical agitation. To see the conglomerates, they are formed of target bacteria stained with acrylic orange. They are yellow or green, and the magnetic particles are red or orange. And you can see that both methods were at least equally effective. But the main difference is that the capturing time for the magnetophoresis approach is 10 minutes instead of 1 hour with mechanical agitation, which is a great advantage, especially for giving diagnostics. Time saving is a great advantage. Here are my references, and thank you for your attention.
Thank you very much. There are some questions or comments?
So during lunchtime.
So what is actually the magnetic field gradient in the experiment? The value, I haven't catch it.
I don't want to give you the exact value, but you can see that
Order of magnitude is fine for me.
the magnetic field gradient is
Yeah, but that's fine. It was simulation, you know. I don't know. I'm just and the size of the particles here.
So the magnetic field gradient is the most parallel here when the
This I
wall of,
understand the color scale, but the order of magnitude is what 0, you know, 1
I don't know how to answer you. I
centimeter. You know, that's fine.
So can you repeat the question again?
So what is the exact number of the maximum field gradient?
Yeah, I didn't make the simulation, so I don't know what to say.
That's fine. We can discuss it.
Okay. Thank you very much. Thank you.
Our next speaker is Andrea Dula, also from the International Center of Biodynamics.
Hello, my name is Andrea Dula. I'm glad to be present at this event and have the opportunity to show you the results of my work team and our collaborators, of which I had the honor to be part. For this project, we focused on viral infection with Botrytis cinerea. This fungi pathogen produces dramatic financial loss around the world. Lactase production is part of the pathogenesis of Botrytis cinerea. Based on this, we developed a fast, robust method to be applied on site on vineyards. We wanted to have a good sensitivity method, able to detect lactase activity on a threshold value of 3 units per mil, which indicates irreversible degradation of grapes due to enzymatic browning. Also, we aim to make it simple to use by operators with minimum training. The principle of the assay consists in taking grapes and crushing them in sterile bags to obtain the must. Then the must was filtered by a PVPV cartridge to eliminate the phenolic compound that would interfere with the detection of lactase activity. A drop of the filtered probe was placed on a piece of paper impregnated with ABTS. The oxidized ABTS produces a green-blue color observable on paper. At the same time, the ABTS paper was coupled with a gold spin-printed electrode, polarized 0 volts. Based on the reduction of oxidized ABTS, we were able to quantitatively correlate the activity of lactase with the intensity of the current. First, the optical detection was studied by taking pictures of the paper sensor with a smartphone camera for different reaction times, different lactase activities, or different impregnating solutions. The pictures were processed with using image J. The optical method based on the ABTS paper sensor showed an increased color intensity with lactase activity in the range of 1.1 to 7.8 units per mil. The 10 minutes measurements were more sensitive than 5 minutes measurements. Next, we focused on the development of the assisted paper electrochemical method. The performed assay had a linear range of 1.1 to 16.8 and a detection limit of 0.43 units per mil for a 5 minutes test. As in the case of the optical detection, the 10 minutes measurements had higher sensitivity with a detection limit of 0.26 units per mil. The dual method was applied on masts from grapes infected, artificially infected with Botrytis cinerea. For these experiments were used four different grapes, four different variety grapes. White grapes, unknown variety, rose grapes, the red globe variety, and black grapes, Moldova, and Tempranillo variety. The berries were inoculated with a 10 microliter solution of 1 million colony-forming units per mil of 3 different types of strains of Botrytis cinerea spores in different incubation conditions of temperature and relative humidity. Despite being of similar size, we observed large variation between berries. The red globe showed the lowest infection rate. The largest enzymatic activity was 9.67 units per mil that was measured in highly infected grapes, black Tempranillo grapes. The white grapes inoculated with the HC strain had higher activities compared with strain 1C for the same infection degree. The value for the HC was 3 times higher than the 1C strain. In conclusion, the paper sensor can be used independently from the electrochemical test for a qualitative 10 minutes control limit test. And it's capable to detect the lactase activity of 3 units per mil. The dual optical electrochemical assay is adequate for the quantitative evaluation of lactase activity in grapes as a fast procedure to screen for fungal attack of Botrytis cinerea. The optical test can easily evolve in the directional smartphone-assisted analytical device to make it an attractive technology for users. Thank you for your attention.
There are some short questions or
So basically, what you want from the technological point of view, you want to have this screen of fungal attack very fast, right? Like with a drone and the miner and so on. So this means that you need to detect, changes in the grapes, very, specifically. What is the time scale? Because your incubation time was, days, weeks, basically. So how this works when you want to detect, on the spot? 24 hours.
Well, you don't need to incubate the grapes because this procedure is made for on-site detection.
But you see this kind of changes, you can optically assess them, how close you have to be, what is the resolution, how you have to fly it, for instance. You cannot go with your smartphone, to check out the grapes. You can do it, but what is the resolution you need to get this, real-time detection, basically.
The resolution of
camera or? Yeah, temporal resolution, yeah. How fast, how often you have to go with the phone on the same grape in the same row of the viner to make sure that, the grape is, safe, is growing okay. You have an idea?
well, I can answer that.
Yeah, that's fine.
Yes. So this method detects lactase activity in mass. You can detect it as fast as the first sign appears on the grape of the development of the cellule. And the method is sensitive enough, but it's not so very relevant for the wine growers. They have this method is integrated in a program for the prevention of Botrytis cinerea now. So what they are doing, they are using these results from periodic testing, and they put it software for risk management together with data from forecast and.
But how often? What is the periodicity of this periodic testing? They do it, daily, weekly?
Not daily. They do yes, weekly on a weekly basis.
On a weekly basis.
Yes, and they have certain locations in the
specific spots, yeah.
Yes,
Exactly.
yes, and they have basically this project, this sensor for lipid was integrated with the GPS
And
sensor.
what type of software they're integrating all this data with?
This is a software that is developed by the Spanish partner in this project. It's not yet
Thank you.
Our next speaker is Raluca Quintano from the International Center of Biodynamics.
Good afternoon. I'd like to present quantitative evaluation of bacterial cells dynamics in a lab chip assay. It is well known that is highly needed for methods for the rapid identification of microorganisms and for rapid SAT, for efficient biotechnology products development, accelerated clinical diagnosis and treatment for treatment of infectious diseases. Our approach was to create a multiplexed high-resolution analysis for rapid, sensitive, and specific identification of microorganisms. We implemented the optical-based electrical impedance spectroscopy assessment with DIC and reflected light microscopy assay for characterization of the phenotypic features of single cells, including the cellular size, motion, morphology, membrane integrity, and electrical parameters, and enable discrimination of persisters from resistant cells upon antibiotic exposure. We take bacterial cells, gram-negative E. coli, were spotted on ETO chips layered with agar strips and investigated in peptone medium with an inverted microscope in transmitted light and reflected light, coupled with electrical actuation for electro-optical label-free multiparametric assessment. This you can see the evolution of E. coli with and without antibiotic. This picture in the left were taken on in DIC in time, and as you can see with high antibiotic concentration, the area of microcolonies are smaller. This method, investigating in time in DIC, gives us 1 hour detection of ACT. These are the pictures seen in DIC here, and these are after the actuation electrical. In the first pictures, they are E. coli at time 0, and these are after 90 minutes of 2 milligram per liter colistin sulfate exposure. We can observe the membrane permeabilization here, here, and here. And we also can see that in electrical obtained images. This bioanalytical platform allows real-time tracking of events taking place at the cellular level and evaluation of the division of the bacterial cells. And it allows establishing the set of representative cellular parameters that quickly reflect bacteriostatic effects towards real-time tracking of cellular effects of antimicrobial compounds. Thank you.
That questions or short comments? If not, thank you very much. Thank you. Our next speaker is Arian Olumi from the International Center for Biodynamics.
Today I will present to you accurate measurements of thin film parameters using the novel SPR system. We introduce a novel common path system, SPR system, for simultaneous high signal to noise ratio, reflectivity and phase angle result assays. By measuring the solutions with known refractive indexes and fitting this reflectivity curve and the phase SPR phase curves with the transfer matrix, we can determine with a high degree of accuracy the parameters and the characteristics of the SPR sensor chips. This is a setup. By using the polarizer at the 45 degrees and the beam displacer, we can both have the reflectivity curve and the phase curve on the camera, on the image. We use the wave plate to adjust the ratio between P and S light so that we have the best contrast in the fringes of the image. Before fitting, we must know the relationship between the pixels on the camera and the angle, incident angle on the surface of the chip. So for that, we make 4 different concentrations of 4 glycerol water solutions, and we use a refractometer to get the true refractive index value of them. I mentioned Snell's law because we use the critical angle at which we have total internal reflection for the relationship between the angle, between the incident angle and the pixels on the camera. These are the data on the pixels. We use a polynomial fitting curve, pixel versus the critical angle, to get the relationship between the pixel and the angle of incidence. By using the calculated relation, we can fit the phase and the reflectivity curves using the transfer matrix method, and we got the following following the parameters which are in agreement with the theoretical values. Over here you can see the reflectivity curve for 2 different solutions and the phase curves for the same solutions. In conclusion, by using the novel SPR system, we can determine thin film parameters with high accuracy, as compared to ellipsometry, where the thickness and the refractive index could be simultaneously determined only if the thickness of the layers exceeds 15 nanometers. This method was successfully used for a 3 nanometer thin film. Thank you for listening.
Thank you very much. Are there any questions or short comments, please? If not, thank you very much.
Thank you.
So our next speaker, also from the International Center of Biodynamics, is Yana Lopez, please.
Good afternoon, my name is Yana Lopez, and today I will present you a part of our work at the International Center of Biodynamics, the optimization of a magnetic actuator of an analytical bacterial detection platform using the finite element method. My presentation is structured as follows: aim and how, the modeling part, the realization of the module, experimental validation, conclusions, and selective bibliography. The aim is to obtain a high value of the magnetic field gradient inside the measurement chamber. The gradient is proportional with the magnetic force. How did we achieve what we set out to do? By simulation, by simulating many different models in COMSOL multiphysics. The modeling part is based on the 5 steps: defining geometry, realization of the physical model, establishing the initial and limit conditions, solving the problem by using the finite element method, and processing the results according to needs. As I said to you before, I simulated many different models, but I choose just 3 to present you. This is the first model that was used in the detection platform. It's based on 4 electromagnets. After that, I simulated a hybrid model with 2 permanent magnets and 2 electromagnets. And last try was a model with 4 cylindrical and 1 spherical permanent magnet. After the simulation, I obtained the distribution inside the measurement chamber. This is the distribution of the gradient for the first model with 4 electromagnets. This is for the second, for the hybrid model with 2 magnets and 2 electromagnets. And this is the third for the spherical model. As you can see, the distribution of the gradient and the magnetic force is better here on the spherical arrangement. After that, my colleagues realized the module, and we incorporate it in our platform. Here is the actuator. The experimental validation. Here is a demonstration. These are the magnetic particles that are moving from one electrode to the other. We are measuring the impedance. And after that, we post-processing the results. Here is represented a period of the amplitude of the signal. With black, we have a concentration of magnetic beads, and with red, we have the double of that concentration. In the second representation, with black are the magnetic beads, and with red are the magnetic beads bacteria complexes. As you can see, the signal of magnetic beads cluster is bigger than the simple magnetic beads. In the conclusion, we developed an optimized magnetic actuator that has better magnetic force, reduce the cost of manufacturing. It is robust and easy to maintain, adjust, and operate. Of course, support of this project is gratefully acknowledged. At the final of my presentation, I want to say thanks to my colleagues. We are such a great team, and thank you for all the things that I learned from you. These are my references, and thank you for your attention.
Thank you. Are there any questions or comments? Please.
So, not you say another question. So you say that you have to be cost effective, but what type of permanent magnets are using this setup? What is the material that the permanent magnet used for this?
I think.
And nothing for Bohr, well, you don't exactly.
I defined the properties, but was 1.5 years ago, and I forgot.
Well, I'll discuss with you guys.
Thank you very much.
In the system, it includes both of the setups to the last I think the first one demonstrated. So when you break, you can see.
Thank you.
I can break it?
No, if you want to stay here longer, yes.
And our last speaker is Emanuela Garbarino from Nanjing Medical University.
I'm coming from the Department of Medical University in Immunology, and I will present the analysis of human herpes 6 and U4 and U26 gene function. Practically, we know recently the pathogen hypothesis is gained more attention and stated that the pathogen have the infectious agents have the active role in the development of Alzheimer's disease. Our study is based in HHV6, and as you know now, after the establishment in 2014, HHV6 is characterized by two strains, HV6A and B. Concerning the HV6A, we know that practically the characterization of Alzheimer's disease are the amyloid beta accumulation and the abnormal tau aggregation in the brain. Beta amyloid is been shown to function as an antimicrobial peptide and act as a pro-inflammatory agent and activate many inflammatory components. But when it's overaccumulated, give the damage to the neuron for the pro-inflammatory phenotypes. Practically, our result, we studied the U4 gene function. It was a gene unknown. And we studied also U3, but the expression was so low, so with the focus on U4 gene. We saw that the mRNA expression started immediately after 8 hours, increased during the HHV6 infection. We inhibit the U4 the expression by express amyloid precursor protein. The amyloid beta practically is produced for the proteolytic processing of APP form. Depending on the cutting and its alpha or beta secretase, practically give the accumulation on amyloid beta plaque. We found in our research there is no direct interaction between U4 and APP. Practically, we suppose that the U4 bind a group of molecules. In this case, it was the E3 calling ring E3. This is a competition between U4 and the APP expression, and so they stop the degradation of the correct degradation of amyloid precursor protein. This is the correct way. We see it here that practically there is a competition with the U4 APP in binding the ring E3. And even the degradation of APP is not correct. Concerning with the HHV6 virus, we know that practically influence sorry, I'm a little bit nervous. Practically, we saw that it affects the immune response as other many viruses. We infect the cell, and we notice that U26 immediately also increase during the infection. And we know that the MAPS is a mitochondrial antiviral signaling protein. They are giving the signal pathway to activate the interferon. Interferon is a molecule that they play an important role in antiviral state. They activate the adaptive response and also inhibit the viral replication. We saw that practically the U26 inhibit the MAPS and also is expressing mitochondrial, leading practically with the potential of the mitochondrial membrane. This is the pathway, and the blocking MAPS, we could see that interferon was not expressed correct. This finding that really the gene, the U26 protein, play a vital role in blocking the correct signaling pathway leading to interferon 1, type 1 interferon, and leading practically the virus replication. I want to say thank you to my professor in China, Wang Tang, and his group, and also my husband, Vladimir Dmitrich, and thank you all for your attention.
Plenty of questions, no problem. Sorry, Chairman, I just want to ask you something. We have to finish the lunch by 2 o'clock.
We finished here.
No, the lunch should be finished by 2 p.m. So we have to start after the session quite soon.
He represents his development of biomimetic structures as building blocks in sensors and actuators. Thank you very much for your introduction and for your invitation to be here. I feel excellent. It's a very challenging environment for me since it's a lot of biology, and biology is not my main field. But biology in the last, let's say, 10 years started to become something that attracts me. And the reason is that I'm from the area, so my background is in, let's say, started in semiconductor physics. And everybody knows that dealing with semiconductors, you need to deal with clean rooms, with things where every defect means it's a drama, and if you build a very small structure, then a defect means that you can throw it away. So it's fascinating how biology doesn't need a clean room to build a processor, doesn't need huge infrastructure to build nanostructures and to build nano devices. So I think it's something from which we can learn, and the methods which are used in biology we can employ. We are far away from doing this, so don't expect to come here with an artificial cell or with something so smart, an artificial neuron or something, but we are starting to scratch a little bit the surface and to try to employ some nano things in dealing with new functionalities. So a little bit the National Institute of Materials Physics. It's one of the institutions in Bulgaria area. So the people who studied physics in Bulgaria, they know because it's in the same building as the Faculty of Physics. And the institute is a relatively large one, about 300 employees, and deals with a lot of devices fabrication with new materials and in a wide area of applications, starting from sensors to drug delivery and energy storage and so on, solar cells. What we also have and use a lot is the area of characterization. Here we have a strong group of electron microscopy, which dates back to the 1950s. So the late 1950s was the first electron microscope installed. Also, we have characterization regarding composition, structure, optical properties, electric properties, magnetic properties. We collaborate intensively with electrosynchrotron. We have now something like, let's say, a third of a beamline there, a Super ESCA beamline with our own experimental cluster, with XPS and related techniques. So it's a relatively large institute, and well, now focusing to the work today. What's important to know is that nanostructures are quite attractive because they are small and you can bring functionalities by changing a lot of parameters, morphology, composition, surface to volume ratio, and so on. But what you need to do with these nanostructures is to employ them in devices, and this is not easy. So you need to have preparation methods which are low cost, which can give you large quantities. And the next step, which is even more difficult, is to employ these nanostructures in such ways that it's relatively easy to use them. So here it's an image which most of you know, that the biological things start from somewhere around 1 nanometer and go to large dimensions, and their typical size is similar to lots of, let's say, biological entities are situated in the area of nanostructures. If we talk here about carbon nanotubes, fulanes, nanodots, semiconductor nanodots, and so on, nature packed a lot more functionality per volume than we are able to do, and probably we will need many years to pack the same functionality in such small volumes. Okay, so you can use nanostructures to build a lot of devices, and you can have sensors, electric generators, photodetectors, transistors, whatever you imagine. You can do this on smaller and smaller scales, and this is quite interesting. A method which we employ to do our work is electrospinning, and this is a method which we started about 10 years ago, and let's say it's similar somehow to how a spider or they build their webs. So but in our case, one uses a high voltage, really high, kilovolts per centimeter. You use a simple polymer solution, and when you play with these two, here you can see that you have let's say it's a syringe. Here you have a droplet of polymer solution, and then you have this fiber forming from this, and this is what you end up with. This is a web of fibers, which are usually 10 times or even smaller, 100 times smaller than what you see in nature. So the textile fibers are somewhere around ranging, so a hair it's about 100 micrometer. Textile fibers can go down in some cases to 10 micrometers. Here you start from 1 micrometer and you can go down to 100 nanometer or so on. So you can play around with small dimensions, but also relatively large quantities, and this is quite important for applications. What we did was to make the next step, because okay, we have a structure which is similar to something which exists in nature, but you need some function to this, and this function can be done using physical or chemical methods, and an example is covering this with gold, for example. So you just do some gold sputtering, and then you can cover this with a polymer, with a conducting polymer like polyaniline or polypyrrole and so on. Then you have a fiber which is both conductive, and a web of fibers which is both conductive but also can be sensitive or electroactive. Here it's how this looks using scanning electron microscopy, and here you can see if you dissolve the inner layer, you end up with some tube which is covered with gold on the inside and polyaniline in the outside. These microfibers, our first application was to employ them as electrodes on paper, and we employed them, we fabricated some heaters. So practically, if you pass a current to this fiber array, you heat it, and you heat it to temperature which exceed, let's say 50, 70, up to 100 degrees centigrade. Here we have some fibers put on textile, put on paper, silver and gold. And what is interesting, you can play around, pulse them and play around with temperature and so on. And
sorry, who's playing on that person?
I think I forgot something from a previous talk. And, yeah. Sorry, I forgot to delete it.
I'm like a cat following.
Sorry for this. I have a newer version with this deleted, but I didn't load the right one. Okay, so the first, here it is an example just to show you how this can be employed. So this is an array of fibers on some paper, and we've written here some things.
and you can see that it's a thermal ink, and you can make it appear and disappear. What is more interesting is that you can use this in, I will show it again, this one. Oh, sorry. Okay. Here you don't have direct contact, but you have RF-induced heating for the area of fibers, and again you can see this heating up. It's a quite interesting application, but we decided here are some simulations. I don't go into details. We just showed up that such an array can heat uniformly the surface. We decided to use this for an application which is, let's say, quite interesting. Namely, this application is related to drug delivery, and we used a kind of sandwich, but we also added hydrogel. The idea is that you can fill the hydrogel with a desired chemical, a solution, and then you heat this and make the hydrogel release the liquid at a desired temperature. So we can control the release of the chemical from the hydrogel according to the desired, let's say, desired dose or something you are interested in. Here, the things are quite related to the previous one, but what I wanted to show is this film to see how you can control it. So here we have methylene blue, and you can see the difference between a heated device and one which is not heated, to see how faster you release the methylene blue from the solution. So practically it's clearly accelerated. We did the measurements, we can go up to 5 times more when it's heated than when it's not heated. Okay, another application which I think is quite interesting is to use the same setup, but this time not for acting, but for sensing. So we covered such fibers with palladium and palladium oxide, and we did some experiments in wearable pH sensor. So practically you put such a patch on your skin and you can detect quite easily. We have also more applications coming on this, but this was our first test. You can detect the pH of your skin, the pH of your sweat. This is quite important because this gives you a lot of data about possible dehydration, but also about other things which may happen, and it's quite interesting to use this, for example, for military or for elderly which need some treatment. They need to be reminded to drink liquids and so on. Here is the, let's say, the usual part with structure, composition and so on, high resolution electron microscope. Here is our sensor. Of course, it's not a very, let's say, there are more steps to be done until you get to the market, but as a principle, this is quite flexible and can sit well on the skin. Here are the data regarding the ability to detect pH, and let's say the properties are quite similar to the ones in commercial devices, commercial pH meters. Similarly, we did a similar again to detect other chemicals, but this not on skin but in cell culture. So practically here we can sense peroxide dismutase, and here you can quantify other ions, so quantify cell oxygenation. This was done by my colleagues. Okay, so we focused on this oxidative stress. As I told you, we use these kind of devices and we can employ it on a rather larger scale. So this webs can be used in a cell culture, for example, in order to assess the behavior of the cell culture, to assess some stress there. Okay, further we have sensing. We have a type of actuation here. We will use a similar arrangement for other types of actuation, and this is mechanical actuation. And I want to show you another film. Here you can see such an array of fibers pulsing when a pulse of voltage is applied. So practically these conducting polymers are also electroactive. If you insert some ions into their matrix, they will increase in volume, and this can move if you. Okay, here it's an example, again an example of electroactivity. Here we placed a piece of wire on such a fiber, and when we give the signal, the fiber lets the wire fall. So again, you can imagine we wanted to test the ability to hold the weight, and the fibers, the webs which were holding the wire, we were not able to weight because they were so light that our machine was not able to register correctly their weight. So it's very, very strong for the size. I mean, this is the direct consequence of the dimension. Everybody knows that spider silk, for example, is one of the strongest materials we can have, and this is an example of how reducing the dimension improves the properties. Here we played around and we did similar experiments to the fibers which were fabricated by ourselves, but in this case we used eggshell membranes. And again we did all this covering with metals, covering with different chemicals, and we found out again that such a fibrous 3D structure can give us a lot of, let's say, help in our application. So a structure which has a lot of active surface. Here we show that humidity triggered movement, so it was a humidity triggered actuation. Again, we did some experiments with things which were moved around. We deposited two types of semiconductors, zinc oxide and copper oxide, or some other combinations, and we formed some very small and very disordered PN junctions, but these PN junctions have a very intense antibacterial activity, and what is more interesting is that they were able to have antibacterial activity using visible light. Usually you need ultraviolet to kill bacteria, but in this case we were able to harvest visible light and to produce some antibacterial activity. So this was quite interesting. And okay, so this is the last part, just another small example of what we are doing. In this case, we deal with some nanowires. We have some various ways of producing nanowires. These are produced by chemical methods, but we also produce them by template replication or by other means. And we inserted these nanowires in various devices. So here you used electron beam lithography in fabricating a field effect transistor. The nanowire is here between these electrodes. You will see some details, and again we played with different types of nanowires, simple nanowires, coarse shell nanowires, and we proved, let's say, photo detection, but also, so here are photo detectors, but also field effect transistor properties. So you can see some of these respond to external, so the conductivity to the channel was tuned by the external potential. So one can employ such a device where you can have it a simple one or a core shell one. You can see here a copper oxide core and a zinc oxide shell. And you can use such a device in order to, for example, to put some devices on the bottom of your petri dish and then have a neural cell culture, and then you can register very sensitive pulses of voltage between the neurons. So this is a thing which we are working to develop. Okay, so conclusions are quite straightforward, that if you want to go to the nanoscale, then it would be a good idea to look at examples from the nature. There are quite a lot, as you saw, the fibers which were doing actuation. It's an actuation which is quite efficient, and the reason for which our muscles are usually also fibrous structures are related to the fact that these are very efficient geometries. So practically, when doing nanotechnology, let's say, it's quite important to look at the natural examples. So yeah, that was, I hope, yeah.
It's fantastic. I think that, well, I like one saying, you know, this is the beginning of a beautiful friendship. You know this. So thank you very much.
Sorry for the anecdote.
No, it's okay. Short question?
Short one or two. So one application that jumps to my mind for the very thin electrodes is for brain activation for implantation.
Yeah, yeah.
And I think Elon Musk is talking about it, but
yes,
many
yes,
others.
yes.
The other thing is, I remember my colleague John Rogers, whose name probably is familiar to you. He's working on deformable electronics. He's working on harvesting energy, so having sensors or other devices in the body that are self-powered from, let's say, from the heart.
Yes.
And harvest electricity. So, do you think it's possible with this kind of electrodes to do that?
Yes. So, for example, one of my colleagues is working on this, on a project like this, where, for example, we have this kind of web electrode system, and at one end this behaves as a sensor, or at the other end this behaves as a battery. So practically one end you have applied voltage from the other end. The other end has some combination, for example, of some chemical reactions which produces you a small voltage which you need to read the sensor. But also one other application, we have a paper which is now waiting to be published. It was accepted. When we made a step forward, you saw that when we were heating an array with an RF field. And I think this is also, so as you have wireless phones and so on, I think one step would be not to need, because to have a battery with this kind of electrode, it will be a problem. But using RF to transmit information and energy, I think is doable, and one needs to do this.
Yeah, I mean, submicron thickness probably is no longer damaging to cells.
Yes,
I think.
yes, you can.
I don't know what the threshold is, but I think it's.
Yeah. We used for the RF, we used something like some kilohertz, and it was quite okay. So I don't think it will be a problem for the living tissue. But anyhow, one needs to work on it. But yeah.
Is there any way of being able to electrospun some magnetic materials?
Yes, we did it already. We did some tests. We do not electrospin only magnetic materials, but you use polymer, in which a polymer solution in which you disperse some magnetic nanoparticles, and you end up with some interesting magnetic properties fibers. So fibers with interesting magnetic properties. So we did already this. It's a little bit tricky because when you have very small particles, then surface tension and all this start to become some problematic, and it's a little bit tricky to disperse these magnetic nanoparticles into a high viscosity polymer solution. But with a little bit of patience, this can be done. We already, also it was one of the projects which we started, but we didn't have enough collaborations to carry it on. But yeah, it's possible, yes.
And about the polypyrrole, how you electrospin?
No, so the conducting polymers are a problem to be electrospun because they are conductive, and when you have something conductive in a 1 kilovolt per centimeter field, then you have a problem. But what we do is these two steps. So first we electrospun an insulating polymer, then we cover it with metal, and then we do electrodeposition. So we do it in a multiple step.
With your permission, I would like to ask you to continue this fantastic debate during the round table.
Okay.
Thank you very much.
Thank you also.
Now I have the pleasure to introduce Professor Sorin Malinte from Belgium, Catholic University de Louvain-la-Neuve. Nano-engineer materials for cellomics, very challenging.
No, it's nice to change my talk today because, next time I'll ask you always to link with your stuff because it's kind of a continuation of what you presented, I guess so. It is not John Rogers' level, don't worry.
Don't worry.
It's not that we cannot do what John Rogers do, it's just we don't have the side.
So everything here is on purpose.
Yes, it's wonderful. So, of course, I like to thank you being here. I like to thank Eugen for inviting me to be present today with you. And what I'll tell you today is just a kind of two small stories. Maybe you heard them. I choose them not because they go all way around to study cells, but I feel that there are some of them in this phase where they maybe look interesting, but then they need, I would say, much more work to be applied. But they have, I think, some features which are interesting, and they are not very popular because they at some point you'll notice that they involve a lot of technological effort and carefulness to do the experiments. But yeah, maybe one can think over. And I know that sometimes when I discuss this kind of things, people would like to have the raw material, which are the thesis with all the details and so on. So don't hesitate to ask me, the details behind the story because they are a bit complex, but you'll see the potential. Okay, so I think it's very simple. Everybody knows today that we have this artificial intelligence, fancy word, but basically plenty of technology which gather around other interests, move on this life science field, and yes, we can say that indeed, if we talk about soft robotics and microfluidics, these are types of things which can help definitely the field of cell analysis. So we are interested in this context in something we call hierarchical three-dimensional structuring. I give here two basic reasons. First, it's important to understand the modeling of complex, I would say, man-made, three-dimensional system, which can be rationalized and used in this field. And then there is, as you already noticed from the previous speakers, is always a demand for, more high-throughput tools, which have this kind of structural complexity to assess, multi-functions, multi-properties very fast and very accurate, okay? And then, of course, if you are working like we work in this kind of engineering facility where we have the ability to construct this type of three-dimensional systems, then yes, you can use this type of three-dimensional hierarchical structure. Sometimes it's referred as nanoarchitectonics, at least the way we are practicing it. And indeed, by using this, you can create these platforms which have very high surface to volume ratio to control really the interface. You can use this type of intertwining, top down with bottom up techniques to really scale up the process. And of course, sometimes the simplest, I would say, output you get are this type of highly ordered arrays of structure we are using for sensing. Okay, and I'll give you some examples today. Okay? So basically, we learned these tricks of building 3D structures like many other groups with nanowires, and we practice this game basically with any type of nanowires, organic, inorganic, hybrid, organic, inorganic. And for instance, here we can use this kinked silicon nanowires. These are as fabricated silicon nanowires by metal-assisted chemical etching. We studied, for instance, internalization of epithelial cells which are sitting on polyethylene methylsiloxane pattern substrates. And of course, you know this field very well. You know that if we are functionalizing this silicon nanowires with peptide proteins which can bind transmembrane integrins, we have this cell internalization tuned up and things like this. We are knowing a bit about this field. We basically focused many times on what we call compartmentalization, and we use this type of combinatorial approaches where we would build materials on top of the other types of materials. And basically we are interested, let's say, in this micro-controlled, micro-patterned environment to do this cell analysis and sensing. Okay? So we have platforms, I would say, on very type of surfaces. Sometimes we use flexible substrates. Sometimes we have integrated all type of sorts, plasmonic, all types of things you already see, types of heaters, actuators, sensing devices. We use often this type of, I would say, soft lithography, like playing with colloids. And if you play with the colloids, then you also start to learn a bit about this microsphere, enhanced microscopy. And we've been working in this field, tangentially, but we are quite aware of what's going on in this field as well. Okay? But I'll not give you details on all these stories today, so I'll just link immediately with what you not said before about this conductive polymer. So here I'll just remind you how we proceed with the patterning of polyaniline nanostructuring. So basically we are using this electrolyzed polymerization of aniline by some catalyst, the platinum here, and then we use this, I would say, complex tool, but which gives us a lot of flexibility if you want to design things, which is electron beam lithography. And then we create this type of nanostructures where we can have this electrolyzed growth of the polyaniline. Now, the fancy thing comes when you are working with the electron beam resist itself. Today I will not go into detail of how we can use this type of template, but let's see a bit. Just one single component resist is what is typically used in fabrication. And basically the idea is very simple. We start with a substrate which can be flexible, rigid, opaque, transparent, whatever you like. Then you just apply a coating of the catalyst, and this is, it's up to you how you want to have it, like thin film structure, doesn't matter, exactly what you want to do. And then, of course, you have this step of building the functional structure. Here is the polymer. And of course, you can coat this catalyst simply with polyaniline, but then you can use this, I would say, high-level techniques like immunolithography to create some specific, structures, arrays on top of this catalyst, and then you come up with something very, very complex, and you'll see some examples. So basically, if you start, as I say, just with one single component, very thin electron beam resist, then you can fill up these patterns with polyaniline, and you see here that you can create these arrays of dots with essentially no parasitic growth in between. So I would say this technique gives you something like 50 nanometer picture resolution. We tried higher, but I would be skeptic you get better than that because the machine we used was a very stable one, a 100 kilo electron volt machine. We collaborated here with Chalmers University, so it is very hard to beat by far this limit if you want to play this game with this material, okay? You start to have problems with the roughness of the catalyst, it will be tough, okay? So this I would say it's a very good result. Okay? You can create different type of structure here, just we use thicker electron beam resist, single one component. And of course here we move from dots to nanowires, to one-dimensional objects. You create these arrays. Basically you see they are highly ordered, they can be functionalized. You can look at them, for instance, at other type of optical instruction like photonic lattices. You can create perfect defects, you can use them in different ways. We have progressed a lot in the past into this field. I will not stay longer on this field today. What I want to remind you is that basic feature that you have some tiny catalysts, which are what we call the reactors. Here is platinum. And basically you can cover this catalyst with the polyaniline with very small amounts, and this is very hard to get with other type of techniques. And the next logical question is, okay, but it works only for platinum. Well, this is what we found out. It works basically with any other conductive material which is connected to the catalyst, okay? And we have this kind of, let's say, combinatorial proof where we have dual catalysts with different sizes of platinum or gold catalysts. They are connected or disconnected, and also patterned with one or different arrays of nanochannels. And basically, the result is shown here. So filled dot means you have a channel defined on the catalyst, and empty dot means that there is no channel defined. And basically, this is what tells you that every time you have a channel defined on the platinum, and this is in contact with the reaction media, will define your structure, but only when the other catalyst is in contact with the platinum and you have a channel defined, you'll get this fabrication done. So this can offer you a lot of possibilities to design where you want to have this, I would say, optical active surfaces integrated into your platform. So we can discuss about this, okay? So as I mentioned, when we play with colloids, and you know that they are used in many ways in microfluidics and in cell analysis. Here, probably you know very well this recent example, where we have this shallow colloids, basically with different dimensions, with the micropore, which basically are mimicking, the spherical membrane of the shell, so you can study, the transport across the membrane. So these colloids are very interesting. The problem, like already Jonas mentioned, when you start to play the game, the point is that you need to control somehow the diffusion in function of what type of colloids we are using, in function of the application we want. And basically you are facing a phenomenon sometimes which is called sedimentation, depending on your platform, but it can be closed, it can be an open microfluidic. And we face this problem also from the point of view of additive manufacturing. As long as you want to play with these colloids and create architectures which are perfectly ordered, you'll start to face this aggregation, this sedimentation. You see here, for instance, materials created which are basically created around the polystyrene spheres with carbon, but you see at the interfaces you are losing this quality of the order you need to control and to create this magic. So
we said, okay, let's imagine how we can monitor this segmentation property. So basically, we are engineers, we look at what tools we have around, so then we said, okay, let's look at coplanar waveguides. So basically we are using microwave frequencies, we are using an object, which is a two-port network, a coplanar waveguide, so it's described by a simple 2x2 matrix which is called scattering matrix, so you don't have to enter into details. This matrix connects the input incident waves and the reflected one, and basically it is described mathematically by the impedance, alternating fraction of length. But if we are matching this network, then this matrix is very simple, and basically we have to monitor only this one single transmission parameter. So basically, this is what we are using, like a device, very easy to be constructed from our point of view. We have here the dimensions. And, I'll not go into detail with the fabrication. It's obvious you are patterning the metal layer with photoidography here. You just need the microfabrication. You don't need a very tough level of fabrication. Of course, you can couple back conductive layers. If, of course, you need optical access. This can be transparent oxides. You have a lot of possibilities. The same how you want to isolate the structures, right? You can use transparent materials. Basically, they have all the time the dielectric constant. It will not change essentially nothing in your. It depends how you want to use it. Okay, I give you what to use, but this can be customized to whatever needs. And in the end, of course, you can couple your microfluidic platform. Here is the most simple one. It's just an open micro container, of polydimethyl siloxane, and there you are performing your study. Okay? So I repeat here again all these details with dielectric constants and array device. And just to tell you that we were very fair, because we will be looking at some nanoparticles. We have a dielectric constant very small compared to water, which is 78. Okay, so if I would have shown you this with titanium oxide or other particles with huge, dielectric constant, it will be different. Here, I want to tell you what are the limits of this technology. Okay, then this can be arranged to give you the signal you want to see. Okay? So this is very important, and basically this is the trick and the difficult part of the technique, that unit calibration. You first have to calibrate basically to match this impedance line, this